Recording in progress. Morning, everyone. Welcome to day 2 of the symposium. So everyone online and people in the audience, welcome. Yesterday was a long session, and the morning session was dedicated to more of the cultural exchange between South Africa and Ukraine, and mainly highlighting Ballinsky's formidable scientific achievements and many other achievements that we didn't know that he had, including playing music and art and so on. Certainly a Renaissance man in the true sense. This morning we continue the program, and today is largely scientific. And to start off proceedings for this morning, we have Professor Alex Gomovsky from the Schmilhausen Institute of Zoology in Ukraine. Also attached to the Stuttgart Naturkunde Stuttgart in Germany, and an honorary member of APES. So it's good to have someone who's been around. I think he's visited us and associated with Ed Bukowski and Isabel Weisby. I got to know Alex through our initial discussions many years ago for the symposium, but it's good to have met him online and good to hear him this morning. Alex will be talking about how parasitoids develop, the notes on embryology and species delimitation in the genus Entodon. Alex, good morning and welcome, and over to you.
Hello, everybody. I'm very glad to present my data and be dealing with such an interesting event as a symposium of Boris Ballinsky, not only as a speaker but also as head of the organization committee from Ukrainian side. It's very pleasant to see everybody, because during the last 2 years, like most of your names were in my agenda, we were trying to combine the program. And now eventually we have this event, and let me share my presentation and share some of my achievements. Is my presentation visible?
It is, Alex.
Okay, thank you. So, my presentation is how parasitoids develop and notes on embryology and species delimitation of the genus Entodon. As Neville introduced me, I'm currently representing 3 organizations. My home institute is the Schmilhausen Institute of Zoology, where I'm head of the Department of Taxonomy of Entomophagous Insects and Ecological Principles of Biocontrol. And currently I'm visiting scientist of the Stuttgart Museum of Natural History in Germany. And as Neville said, I'm attached to South Africa and to Wits University as an honorary researcher of the APES, of the School of Applied Ecology. So I'm not comparing myself with Ballinsky, but I think this is some kind of coincidence that the home institution of Boris Ballinsky, which is my institution currently in Ukraine, and the institution where he eventually developed his prominent career as embryologist and zoologist, somehow linked to my career as well. So this was also a great inspiration of my scientific activity and actually of this presentation as well. Returning back to the personality of Boris Ballinsky, we know them well as embryologist, as one of the stars and actually one of the founders of the modern day embryology. But as it was stated in the previous day, Ballinsky was also a prominent entomologist, professional entomologist who was dealing to larger scale with Lepidoptera, but also other organisms. And his collection was donated by him to the Natural History Museum of Ukraine in Kiev, and one of he was dealing with wide spectrum of butterflies and moths, but one of the key objects of his research was Acrea horta, the quite common butterfly in South Africa. He was studying the wing pattern diversity and some biological aspects of this butterfly. What is interesting that his rigorous studies on this butterfly also resulted in some observations on its parasitoids, which was also reflected in the paper of Claessens. It's underlined here that actually Ballinsky's studies pushed also an idea of studying parasitoids of this poisonous butterfly. And we can see that despite Acrea horta is considered venomous, its caterpillars are feeding in Kegelaria africana and they contain cyanides. Nevertheless, whatever poisonous insect is, there are always parasitoids and parasites which can overcome this poisonous reaction from the host. And here some pictures which I've done in Northcliffe in Johannesburg of the parasitoids of Acrea horta, Brachymeria cassellensis, and there are also some new data on its biology which are still to be published in future. So we can see that whatever at least insect objects we see, there are always parasitoids behind, whatever it's venomous or hidden living or whatever. So parasitoids are everywhere. And this is actually the object of my studies. Just to remind who are parasitoids, they are tiny hymenopterans, tiny wasps, which lay their eggs either inside of another insect, which is called host, or outside, and then the larvae of these parasitoids start slowly eating the host, and they employ different development strategies. Sometimes they have symbiotic viruses, sometimes they have chemical approaches to suppress immune response of the host. Sometimes there are interesting genetically determined development strategies, like for some polyembryonic parasitoids, the excessive amount of RNA makes larvae to be soldiers which fight with other parasitoid larvae, and they die themselves afterwards, while the larvae having no excessive RNA, this is actually the picture of there, they develop normally. Yeah, there are some parasitoids which suppress their rivals, like here chemically. Some of them suppress the rivals of being their own siblings mechanically, and so on. And then the winner comes out of the host, like exactly like here, like in the horror movies. So I think parasitoids are quite interesting to be studied. So my research objects are one of these parasitoids. They are species of the genus Entodon. They are tiny parasitoid wasps, and they are associated with a wide range of beetles, mostly of weevils and bean beetles, but some others too. And their hosts are either hidden or exposed. The larvae of beetles can develop inside of the plants or living outside. And these parasitoids can attack either larvae or eggs. And the types of this parasitization can be very diverse and range from solitary, when one egg is laid in the host and then eventually one parasitoid emerge, or gregarious, when many eggs may be laid and all of them develop in one and the same host and then emerge. Actually, this will be the point of this presentation. So if to illustrate what I said in the last sentence, these are some examples of the parasitoids, how they do. So some of them lay one egg in one host, and some of them lay many eggs. And this host may be either egg or larvae. And in case of egg, these parasitoids start developing in egg and develop very slowly, so they start what we call destructive feeding, so killing their host when it reaches the final stage, final larval stage. And if they lay eggs in the larvae, then development is a bit quicker, and then the development starts in larvae and ends up in the larvae. And there could be one or many parasitoids, and they interact somehow. So how do they interact? When they are solitary by name, then they behave like the Highlander from the famous movie. So there can be only one survives, and all the rivals are killed. This process is called siblicide. So they are absolutely intolerant, and when they find anybody else in their host, then they kill the rival. Alternatively, gregarious parasitoids, they are very tolerant by default. Many eggs are laid in the same host, then larvae emerge, then these larvae don't kill each other, they are tolerant, and then all of them get to the final development stage. They become adults and come out, So they are tolerant. And there are many questions, at least two main questions arrive from this point. So are there any cues that the gregarious parasitoids employ to avoid aggression? So why solitary kill each other and gregarious are tolerant? What are mechanics of this process? And then if gregarious parasitoids are tolerant, then are they tolerant only to their siblings, or they are tolerant to everybody? And these were the key questions I tried to answer. Actually, if we look at the ratio of the egg larval on larval parasitoids, then I devoted like 10 years of my career on studying egg larval parasitoids, and these pictures which I have demonstrated before persuaded me in that they are engaged in siblicide, they kill each other, only one emerges. While the larval parasitoids, and especially gregarious larval parasitoids, they were less commonly studied, and there was no idea about interactions of these larvae. So I will demonstrate my achievements, and will try to link somehow it to the ideas of Boris Ballinsky based on three stories. So one will be based on the bean beetle parasitoids, which was introduced from Africa to Hawaii. Another is entirely African parasitoids of bean beetles, and then the parasitoid of weevils in Europe. What they share and what is different between them. So the first intriguing story. It started from that somebody who did like natural jewelry. Accidentally, probably, we reconstruct this story, bought a bracelet like this with bright, colorful seeds of the coral tree, African coral tree of the genus Erythrina, and brought it to like this jewelry, which can be seen in many shops in United States. So he brought something like that from Africa to Hawaii. And it happened to be that these seeds were not treated. They contained living larvae of African beetle, Specularius impresithorax, and this beetle was introduced to Hawaii. And Hawaii has Hawaii have their own coral tree, the endemic coral tree called Erythrina sandwichensis, the common name Willy Willy, and this Willy Willy is a symbol of Hawaii. But the beetle didn't know about it. It just has found that it's very tasty tree, probably more tasty than the African tree it's normally developed, and it started devastating the trees. It started eating the seeds, so how like it's on the picture, but luckily, incidentally, some of the larvae were parasitized, and the parasitoid was introduced to Hawaii as well. So my colleagues from Hawaii sent me the parasitoids and this intriguing story, and saying that what is it? And it happened to be a new species, but the biology was quite problematic, because I instructed my colleagues from Hawaii how to dissect the host and to isolate larvae, and only mature larvae, only the big larvae were submitted to me. So they said that they cannot find the young larvae. It made me to come to Hawaii. Hawaii are very picturesque, but apart from the nice nature, we were like locked in the Hawaii Department of Agriculture. We made like here the temporary culture of the beetles and the parasitoids, and we tried to trace the biology of it. This is actually the beetle. This is what it does. It lays eggs directly on the surface of the dry pods, what is quite useful when you have the laboratory culture. You can have dry pods in the beetles, and you can have the culture. You can have beetles in the captivity. They lay eggs, and then the larva emerges and then burrows into the seed and eats the seed from inside, and then pupates there, and the beetle comes out. When we started playing with this culture in the Hawaii, there was an intrigue, because I also couldn't find the first instar larva. Well, there was a batch of eggs. When I dissected the egg, the host egg of the beetle, there were eggs of the parasitoids. There were many of them. And then there were some kind of fat granules, and that's all. And I understood that it requires more precise studies. So the culture of the beetles and the parasitoids were taken to the Zoologische Staatssammlung München in Germany, in Munich, where I had my fellowship by that time. Actually, Munich is one of the cities where Ballinsky started his journey from Ukraine to South Africa. It was the city where he was resided for a while. Okay, when I was in München, I was trying to discover the biology of the new species of Entelodon. We provisionally named it Entelodon erythrina. It was new, but the biology was completely ununderstandable. And after series of experiments, what happened to be? Actually, in all insects and parasitoids also, the egg produces an embryo. Embryo develops inside of the egg, and then it forms the larva, the so-called first instar larva. It comes out of the egg, feeds, and molts and becomes second instar larva, third instar larva, and so on, so on. And the first instar larva at least in Entelodon, it's very recognizable. It differs from other instars. And what we have in case of this Entelodon, and why I think it's related also to Boris Ballinsky studies, because it's directly linked to the embryology. Actually, inside of the egg, no direct embryo is formed. Instead, there is a proliferating mass of cells which enlarges in size, while the external layer of cells, it hardens and stretches. So eventually we have a batch of eggs. One of the egg starts developing. Cells are proliferating, but they represent just a mass of cells, undifferentiated mass. And then this mass becomes larger, and they stretch the external layer cells so we can see them. And this is what I call the soccer ball chamber. So it resembles to some extent the soccer ball. And inside there is a mass of cells which directly develops into the second instar. So I say it's like if in humans the pregnant woman would give a birth like of a teenager, not a newborn child. Yeah, so directly the development omits the first instar and already second instar forms. Then the second instar larva leaves the chamber, starts feeding, and then development goes as normal, the final instar and pupation. So the peculiarity of Enthedon erythrina is the lack of the first instar. Actually, this study was published, and apart from the scientific value of the discovery of this biology, it was quite successful biocontrol program. Enthedon erythrina was one of the biological control agents which allowed to suppress the pests of erythrina, and it was an action of saving Wili Wili, yeah, saving of the symbol of Hawaii. So this is an example how blue sky science can contribute to applied science, and they can be nicely combined for the welfare. So it was quite intriguing, and it was a part of it was a sort of continuation of my previous studies on the Enthedon parasitoids of the bruchid beetles. But it was quite important to understand whether all African Enthedon parasitoids of bruchid beetles are similar to Enthedon erythrina. Do they all have no first instar? Do they have this soccer ball chamber and mass of proliferating cells at the first stages of its embryological embryo? Or maybe this is the only case, the Enthedon erythrina. So I had to find bruchids and their parasitoids, and it was quite a difficult way. I started in collecting it in different countries of Africa, collaborating with African colleagues. So, for example, this was in Senegal, and in collaboration with colleagues from Dakar University, Sheikh Anta Diop University, and also the colleagues from Institute of Development in Dakar. They have also the cultures of bruchids. This is how they look like, and here in the yellow circles we can see the cocoons of bruchids. When the larvae come out, they make such cocoons. And also in collaboration with colleagues from International Center of Insect Physiology and Ecology in Nairobi, in Kenya, I have observed and collected many bruchid parasitoids, but the problem was that not many of them were cooperative enough. Yeah, many of them were dealing with fresh pods, and they oviposited in fresh pods. So when you collect these fresh pods, they start decaying, the beetle larvae die, and you cannot trace the entire development in the laboratory. And then a good chance appeared. I got in contact with people from Wits University, in particular Isabel Wiersbe and her team, who were dealing with plant rehabilitation and phytoremediation of the contaminated lands. And these plants which were observed or used in these programs, one of them were small acacias, yeah, now what we can call in the broad sense acacias, and one of them was the Dicrostachys cinerea, the sickle bush, very common plant in Africa, in South Africa in particular. This is very nice and very common, very attractive plant. You can see it's flowering, then producing small buds, then the buds develop, and then they have this very characteristic bunch of beans, of pods, and when one collects these pods in field, there are lots of tiny holes, which suggest that the bean beetles developing inside, they are nicely parasitized. But they parasitize by what? They parasitize by parasitoids by default. And this is nice. Also, what is necessary for experimental biology is necessary to have a model object. So, unlike other bean beetles, but another insects affecting the pods of acacias, here it was nice model system of the bean beetles of the genus Broochidius. You can see they lay eggs on the pods of on the ripe pods of sicklebush of the Dicrostachys. Then larva develops. Then likewise, in the situation of the Specularius impressithorax, it eats the seed from inside, then pupates, and then the adult beetle comes out. So actually, it provides you with the possibility of playing with this laboratory culture of beetles and parasitoids in the captivity in the laboratory. And what happened? That there are another parasitoid, another egg larval parasitoid, who is interested in eggs of beetles. You can see the female finds the egg and then it hooks its ovipositor, or we can say sting, in the egg and starts drilling it and then lays eggs. And what I have found, I expected it to be the same as Entodon erythrina. Then there are egg, then egg develops in the proliferating cell mass sealed in the soccer ball chamber, and so on. And no, the development happened to be absolutely same as in solitary parasitoids of other beetles. So egg produces first instar larva, then it develops normally, then pupates, and so on. So no soccer ball chamber, and the African bruchid parasitoids, the African Entodon parasitoids of bean beetles are not the same, and they can have different developmental structures or strategies. Okay, so these expectations failed, but it was still a challenge to identify who is the parasitoid of this beetle, and what is the necessary part of the work of entomologist is the work with museum collections. So you can see the diversity of the specimens emerged from the pods of sicklebush, from small to large, with different degree of the darkening of the tibia of the hind legs. So it took some time to go to London, to the Natural History Museum, to study the types, and again, back to Ballinsky. Ballinsky also had his stop in London in his career on his way from Ukraine to South Africa. He was studying and he was working on embryology, but we keep in mind that he was entomologist in his soul and then also professional. So London is probably one of the most important places where entomologists have to spend some time, and in particular studying the type materials for proper identification of the materials which he or she got during laboratory studies. And then we go to the third story. Okay, what we understood? We understood that some egg larval parasitoids have a different biological strategy, and that gregarious parasitoids can have a soccer ball chamber and proliferating number of eggs and omitting the first instars. Okay, what about the larval parasitoids? This was about egg larval, when they lay their eggs in the eggs and then develop in the larvae. And there are some parasitoids which lay eggs in the larvae, already in the larvae. Do they have the embryological peculiarities and development strategy same or similar as Entodon erythrina? This is what I wanted to see by studying in the European or Palearctic parasitoids of beetles. There is a group of entodons which are called Sioni species group, which includes 3 species. Here they are in the picture. They're quite distinct. One has entirely pale hind tibia, 2 have partly darkened hind tibia. One is very slender and with long antennal joints, with long guster. Another has everything robust, short antennal segments, short guster. And they are parasitoids of Sionus weevils, which have externally living larvae feeding on figwort and mulleins, the scrofulariaceous plants. Here in the left upper corner, you can see some short video made by me years ago. This is how they developed. You see the female searching for the larva, the host larva, and then it rides it and lays eggs. And the problem was that actually only field collected females were suitable for such experiments. Because when you rear these parasitoids in laboratory, you can see that nicely fed, mated females, they basically are afraid of these beetles. This is an understandable phenomena. I tried different approaches to persuade them to do to attack the host, but you see they keep distance. So it was necessary to stay in field for a long time, waiting when the female of the parasitoid lands on the plant, then collect it in the field, then bring it, then to make the laboratory culture and so on. What was the key point of the research? I tried to check first of all the biology. So the females were laying eggs on the laboratory grown larvae, so they were not parasitized for sure. They were newly proposed, and then to dissect and to observe the larvae and eggs. And when you dissect the parasitized larva, you can see the eggs. And what kind of results I got? Yes, the parasitoids are parasitizing the same host. They very minimally differ from each other, mainly from the body proportions and color, but actually the eggs are different. You can see there are some external envelope in the Entodon Cionobus and no such external membrane in Entodon Zanara. Probably this envelope, the external membrane, facilitates attachment to the guts or internal organs of the weevil, but another species manages it without such an envelope. So but anyway, the morphological differences in eggs is obvious. But what about larvae? Well, larvae do not differ, and actually they do not differ much from the solitary parasitoids. The only difference is they are moving a bit slowly, like lazy or sleepy. So actually, what is interesting that basically there is a simple way to avoid aggression. There is not necessary to make such a complicated embryological and developmental strategy as Entodon erythrina, and I believed before that this is the only way to avoid aggression, to avoid first in star, which is always aggressive and killing rivals. But there are another way, simply to have larvae which are lazy, which do not bite each other. So sometimes there is a simple evolutionary decision rather than complicate embryological combination and relocations. And then what about behavior? Does behavior differ? It happened to be that these species which attack same host, they have quite different behavior during their oviposition. So you can see that in Entodon zanara, the 4 legs are put on the host, while in Cyanobius, it tries not to touch the host with the four legs. You see, it oppresses it like dog when serves. Yeah. While Zanara doesn't have a shame of having its four legs dirty on the surface of the host, and the host is generally covered by slime. So species are very similar, but have very tiny differences in their biology. Either eggs are different, or behavior, or both. So these are the main conclusions from this part of experiments. How they minimize aggression: simply do not bite each other, nothing else. And is embryonization is an ultimate way to avoiding side-by-side? No, it's just a contribution likely to delayed development. And whether ovipositing behavior and morphology of immature stages differ? Yes, it does. And then, okay, it was about each species separately. And what about if we combine them? How do these tolerant parasitoids interact with each other? Do they hate or they like the representatives of another species? So for this purpose, I tried to parasitize the same host with two different females, either from different species or from the same species, but two different females proposed the same host. And then to dissect them, or another part of experiment, to trace what will be eventually. Who will win, actually, if two different species are combined? So actually, if the parasitoids have parasitized the host larvae with at least 1 day intervals, for sure the one who parasitized first was growing quickly and they were the winner. So it was a very simple part. But I also tried to persuade the females to sting the same host nearly simultaneously. They tried not to do it, but I persuaded them indeed. They tried not to sting the host which was stung exactly recently by the female, but after at least 40-30 minutes they can do it. And then we can see the results. Some of the hosts didn't survive such experiments and died, and some of them failed to create the cocoon. Normally they create the cocoon, but the parasitoids were developing inside, and some of them produced the pupae and adult parasitoids. And what was the result? The results was quite surprising. So some of the hosts died, but from the survived hosts, were quite strange results. So when I combined Enterodon Cyanobius female and Enterodon Zanara female, and they stung in the same host, then either only Zanara emerged, or Zanara and Sioni, but the host female was Cyanobius, but we have Sioni. And then I tried to make a combination of two females of Cyanobius, and in Cyanobius never emerged. Instead, Sioni emerged, which was not the host species. So actually, one can say that one species gave birth to another species, as here as well. And this is a very simple conclusion, that these two, Cyanobius and Zanara, they are true species, and Cyanobius is just a derivation of the Sioni is derivation of Cyanobius. So when the conditions are harsh, when we can see the superparasitism, then there are excessive number of larvae develop in the same host. Under this stress, Cyanobius transforms to Sioni. So these are slender form. They are this is just a feedback from the harsh conditions. So this is just morphological modification. Despite this species was described nearly 200 years ago and was included in all revisions, and we can see that from a morphological viewpoint, it's very distinct. Nevertheless, if you don't know the development and biology, you can have such conclusion that this is a valid species, but indeed this is just a super parasitic form. So some conclusion that larval interactions define whether species can be solitary or gregarious. So whether it is tolerant or aggressive, it's the background of being solitary gregarious, not actually the number of eggs. And when solitary are involved in sublicide, then the gregarious are obliged to be tolerant. And there are many ways how to avoid aggression on the larval stages, either by embryonization or simply by slow behavior of the larvae. And gregarious parasitoids can demonstrate differences as well as in the ovipositor behavior as in egg morphology. But larvae are the same. And gregarious species are poor competitors, so they avoid competition simply by ignorance of hepatoparasitized host, as I said. But if they fail, then they offspring meet and they produce morphologically very different offsprings, which can cause taxonomical problems in parasitoids, because these superparasitic forms have very distinct morphology. So parasitoids are very tricky. And thank you very much for your attention. I'm very thankful for all colleagues who helped me ever in my life, and especially people from South Africa, the organizing committee, and colleagues from WITS. And again, you for your attention. Thank you very much.
Thanks very much, Alex. That was very fascinating, especially the last bit, which was a bit of a surprise in terms of the change in morphology. We've run out of time, but I'll take one question from the audience if there are any. There's one question, Alex. I don't know if there's any online. No. All right. One question.
Okay. Hi, everyone. Thank you for the wonderful talk, Alex. So my question is, you mentioned in your conclusion that the gregarious species were very poor competitors. So I just wanted to know, when you compare the more solitary and the gregarious species in terms of the parasitism, which one is more successful when you consider, when you look at when you look at parasitism in general, are the one pest more difficult to deal with than the other when you compare the solitary and the gregarious?
Thank you. Very interesting and intriguing question. Actually, once they still do exist, they both are successful. So it's to some extent related to CAR and R strategies, because solitary they produce one offspring. Each egg produces one offspring, but it is a fighter. And gregarious, you see, they're tolerant, but they produce more offsprings, and there are more chances that any one of them survives. And for sure, if there is a chance when solitary meets gregarious, then solitary will win, because it will simply kill gregarious one by one. There are some cases when different parasitoids meet and solitary kills the gregarious rivals, even if they are congeneric. But in larger view of the parasitism strategy, it all depends on the host. So actually, sometimes it's more feasible to be gregarious. Sometimes it's more feasible to be solitary. Actually, solitary parasitoids generally parasitize the hosts which are more available in nature. So there are more hosts and more rivalry. The more rivalry, the more stronger you have to overcome the rivalry. And gregarious, they actually quite often they parasitize the hosts which are more dispensed and occur more dispersely in the nature. So once upon a time the female finds the host, it's more feasible to lay more eggs and the more offsprings come out, because probably while she will be searching the next host, it will be eaten or killed. So she uses its chances. So it's apart from the developmental strategy, it's also ecological issues, whether the host is available. And then there are also the points of the sex allocations, how many females, how many males can emerge. You probably know that parasitoids can manage the sex ratio of the offsprings. They can fertilize their eggs selectively, so actually female, if we can say this anthropomorphic term, decides whether she wants to have more sons or daughters. This also defines whether the solitary or gregarious strategy will be successful. But yeah, this is big chance of nice studies if you start dealing with it. Okay.
Thanks, Alex. That's all we have time for. But thanks again for a very interesting and intriguing talk on the parasitoids. Thank you very much.
Thank you.
Okay, our next speaker is someone who's familiar to all of us at WITS, so certainly. And I was asked to introduce him yesterday at the talk at lunch, but I get a chance to do it this morning. So it's a great privilege to have Professor Marcus Byrne from WITS University to talk to us today. Marcus, I was intrigued by your title, but I did see the play on words on this, and that's very important. From the dung ball, everything, and you might recognize that related to the, I think it's with the egg, right? Yes. But Marcus will tell us about that. Our evolution has shaped the ecology of dung beetles, so it's a great privilege to have Marcus Byrne this morning, and I'm going to hand over to you.
Thanks, Neville, and thanks for coming along, folks. Hopefully I'll be able to entertain you with something slightly different than what I showed you yesterday. But what I want to do is firstly pay homage to Ballinsky in two ways. One is the selection of an ideal model organism, and he found his, I think, in Xenopus, the clawed toad, clawed frog, and did so many embryological experiments on that animal. And we have found that the dung beetle is an ideal experimental animal in that respect is a model animal for asking all sorts of interesting questions. And what I want to do is show you some interesting work that we've done at WITS on dung beetles and using dung beetles as a mechanism for asking questions about evolution and the environment. So Neville's right. The phrase is from the frontispiece of Harvey's Treatise on Animals, and this phrase is ex ovo omnia, everything from the egg. And it's a beautiful phrase because it describes how literally every animal comes from the egg, and I've twisted that a little bit because I'd like to use it as a metaphor that the dung beetle and the dung ball, this stuff here, can be considered like an egg, and so many things can emerge from it, a living animal, but also all sorts of interesting conclusions and questions. So the dung ball is an incredibly important item in the life of a dung beetle because it is a vehicle through which it will carry its genes into the next generation. And so this, fairly foul structure gives rise to the animal itself, but also to questions about how it gets there and manipulates it. So a little bit of basic biology on dung beetles. There's the egg that's laid into this food package. So that's what this is. It's a food package for the larva. And the larva completes its complete development inside that, effectively a lunchbox that mum or dad or both have provided for the larva. It pupates inside there and emerges as an adult. And this allows us to manipulate the environment that the larva develops in, because this food package is everything for this animal. It's like you getting stuffed into your lunchbox as a school kid, and that's the only lunchbox you're going to get for your complete development, and your mum puts the lid down and she shuts you in and sends you off to school. And the manipulation that we can do here is we can change the food source, we can change the amount of food, but we can also change the gaseous environment that that organism develops in. And the obvious gas to manipulate is carbon dioxide, because carbon dioxide has changed dramatically over the last 200 years since industrialization, from a fairly stable state of around about 200 parts per million, and we're now looking at 400 parts per million. And what we did was we reared this South African dung beetle under these different conditions of elevated or, in fact, reduced CO2, and looked at how the animal developed under those circumstances. And what we did was went back in time, so we went to pre-industrial levels of carbon dioxide, so about 250 parts per million. Our current level of CO2, about 400 parts per million, 600 parts 30 years into the future, and then 800 parts 50 years into the future. And so the result we got was actually quite upsetting, because what has happened is that over the years, and this has already happened, we assume, that the dung beetle size has actually reduced significantly from the past pre-industrial levels of CO2. And if we look into the future, that animal size is reduced even further. And what's even worse is that the survival, the probability of survival to adulthood is also reduced at these elevated levels of CO2. And we think that it's actually mediated through the microbiome that the larva is living in, because this dung ball is effectively like an external rumen. Imagine a cow, the way it digests grass, it has a whole load of microbes living inside it. Well, this animal has to eat effectively the same material. In fact, it's waste grass, and it incorporates a whole load of microbes into the wall of the ball, that it goes around and it eats the wall of the ball, and then it defecates onto the wall of the ball, and then it re-eats that substance. And we think it's the microbes that are getting affected by the elevated CO2. Interestingly, the microbiome of the larva is very, very different from that of the female and also from the male. So we think this microbiome here is very critical for the development of the larva, and that's the research that we're carrying on with at the moment. Okay, so in that regard, we can consider dung beetles as a sentinel, as a canary in the gold mine or the coal mine, telling us what we are doing to the environment and what sort of effect it's going to have on other animals. But if we go above the ground and look at the more typical behavior of ball-rolling dung beetles, this is what we typically see beetles doing. And this, again, makes them an ideal experimental animal, because they don't care. They are absolutely oblivious to us. They are quite happy for us to move around them and manipulate the environment around them. And all the time, they just carry on with their work in this determined way. And that makes them an ideal experimental animal to ask all sorts of questions about, why are you doing this? And the other question is how are you doing it? So The first question is, why are you doing it? And we think it's because of competition. This is a rhino dropping covered in a single species of beetle. This is Pachylomerus femoralis. There is clearly massive competition taking place at the surface here, but also underground, actually 80% of the beetles are tunneling species. They're not rolling species. And in any given dung patch, you can get up to 100 species of dung beetles. So there are as many dung beetles packing that dung away underground as there are on the surface of the dung that we're looking at there. Okay, so the question then is to ask, why do you want to roll your takeaway lunch away? Why do you want to roll it across the environment? And what we've done here is we've marked every position of the beetle approaching the arena with a green dot. And then as it tries to depart the arena, we've marked the fight or an encounter with another beetle with a red dot. So not only are these beetles avoiding competition with the tunneling species underneath the dung, they're having to fight with their colleagues over the poo that they're trying to roll away across the landscape. This is a highly competitive environment in which these beetles are working. So, how do you avoid that sort of competition? It's going to happen outside there at the lunch table later this afternoon as you grab your croissant and you head off in a particular direction with your little food package. The best way to get away from your competitors is in a straight line. And that's exactly what we're asking the beetle here. Do you know where you're going? Are you going in a straight line? And that's exactly what these beetles do. Here's the ball approaching the ramp. There's the obstacle that we put in its way here. So there's the approach angle, there's the departure angle, and what we've done here is plotted the mean departure angle, and the average is about 1.2 degrees. Those beetles know exactly where they are going. And so the question is, how do they know where to go? And this behavior is a giveaway, is that at some stage during the rolling behavior, they climb on top of the ball and they do a little 360 on the top of the ball. We've called it the dung beetle dance. And we think that they are looking at the sky. This guy's got a hat on and he's looking pretty confused. So what are they looking at in the sky? Well, the obvious cue, the celestial cue that they would look at is the sun. And so what you've got here is Marie is holding a mirror, and she's effectively directing the sun from the other side of the sky. And Basil's got a shade board, and watch what happens when he shades out the sun, she puts in the mirror, and the beetle does a 180-degree turn. Clearly, the animal is looking at the sun as a celestial cue to orientate itself across the landscape. And because these animals are so malleable and so cooperative, we are literally able to repeat these sorts of experiments over and over again. This is with the same individual, and end up with these sorts of data that we can then analyze the mean angle of rolling under the sun, the mean angle, rolling under the mirror, and literally come to firm conclusions about the behavior of these organisms. And we've done literally 20 years of work on this orientation behavior, and we've worked out there's a hierarchy of cues. At the top of the cue system is the sun. There's a polarization pattern in the sky that you and I can't see. There's an intensity gradient that you will actually be able to see if you go out today, because it's a clear day. You'll see the top of the sky is darker than the horizon. There's a spectral gradient. You won't be able to see that, but where the sun is, the sky is greener. Trust me, I'm a scientist. And we've even shown they've got a wind compass. So what we've got here is a is a fallback system of orientation behavior that if one cue is not available, then another cue can be used by the beetle. What's interesting about this is I told you that you could find 100 species of dung beetles under one dung pat. In South Africa, there are about 800 species of dung beetles. So this is a massively competitive environment. Of those 800 species, only about 70, so about 10%, I've always been bad at maths, Nick and Siobhan will attest to that. But anyway, about 70 species roll balls during the day. They are diurnal species. And there are about 10 species that roll balls at night. And again, we think it's because of competition, because there are these celestial cues that are available to them, and what they've done is avoided the diurnal competition by rolling their balls at night. So I'd like to then jump to back to a diurnal species. I think this is a beautiful beetle. It's called Sisyphus. If you know your Greek mythology, you'll realize what a cool name it is to give to a whole genus of dung beetles. They're called the spider dung beetles, as you can guess why, because they've got such long legs. And the cool thing about Sisyphus is that he, and it is normally the he, rolls his ball that way. This animal gets up on its back legs and it pulls the ball towards itself. Why does it do that? Another question. Well, you know, when you've got a takeaway meal, if you can incorporate a partner into your foraging behavior, well, you can both go home together, dine out on the takeaway, and who knows what event might eventuate after you've had a meal together. And so we've investigated this behavior because it's an incredible behavior, because these two animals are collaborating to go somewhere where they don't know where they're going. All they're doing is orientating to a celestial cue and then finding their way across the landscape. Who's driving? Well, the male has just been removed. Sorry, girls. Put the male back, and off they go again. So it could also be a sexual selection behavior, that you're checking out your mate, what their abilities like at moving across the landscape, moving the dung ball together. Or it could be that it's also advantageous within the habitat that you live in to work together. And Sisyphus is typically found in forested areas. And in forested areas, there's lots of leaf litter on the ground. There are twigs and other obstacles that need to be climbed over. And it would appear that the female is of value in moving over these obstacles. Because if we look at the data here, and we've done this with two species of Sisyphus, Sisyphus fasciculatus is a South African species. The pair can climb much quicker, or significantly quicker, than the solo male. The female won't climb on her own. And we did it with a European species, and we found exactly the same result. So this collaboration is going to be the subject of further research, because just how these two animals communicate is interesting, but in themselves it's interesting that they can cooperate in this way. So I've described the diurnal species, I've made reference to the nocturnal species, and really all there is to say here is that we've worked on a nocturnal species of beetle that uses the Milky Way as its orientation cue, which in many respects is a very romantic thing. So we've added stars to the orientation cues in the nocturnal species. And this really grabbed the attention of the general public all over the world, literally. We got poems and letters and all sorts of stuff. People were inspired by the idea that this tiny little dirty beetle was using the center of our galaxy as its orientation cue. The problem is what effect are we having on these animals in these, you know, it's a romantic view of these animals. We love them very much. They're doing this wonderful thing. But what are we doing to them? What does the sentinel tell us about their behavior in this situation? So you'll recognize this as one of our arenas. There's a dung beetle sitting on the dung, on its dung ball in the middle of the arena. And you might recognize this as the gate out onto Empire Road at night. So this is literally done at Wits University. Downtown Johannesburg, how is an animal that orientates by the stars going to cope with the city lights? And it's not just one species of dung beetle. There are literally thousands and thousands of animals that move around our planet at night, and we have got no idea what effect our night lights are having on them. It's called ALAN, artificial light at night. What effect is this having on hordes of animals moving around at night? So this is Joburg when we don't have load shedding. So again, the animal is acting as a sentinel, is acting as a pointer for many, many other species that have to cope with these environments. So we did what we thought was a spectacular experiment. It was great fun to do. We had one portion of the team was in the bush out at Bella Bella, and the other portion of the team was literally on the roof of the OLS building, the biology building at Wits. And the guys in the bush could see the Milky Way, and we were on the roof of Wits University, and we couldn't see anything because of what's called sky glow. The upwelling light from the ground below us strikes dust particles and moisture in the atmosphere and literally reflects back, and it obscures all of the stars in the sky. So what was the result? Out in the bush, no problem. Normal dark sky, the beetles could disperse. In the city, where there's light pollution, what we found was that they basically orientated to the nearest bright light, and we call this beaconing. So that in itself is not a terrible thing, but nevertheless, what it means is whatever animal is orientating using this cue ends up in the same place. And that clearly is an oddity. If we block out the beacons and we leave them only with this polluted sky, then we get complete disorientation and the beetles are literally lost. And I think this is really sad because the night sky and astronomy is our first science. It literally is the first science that humans ever engaged in, and we're losing it. There's 30% of the planet cannot see the Milky Way. Every single culture has a mythology based around what we see in the sky. And these things are now disappearing from us. And again, we need to think about what we do about this. So, to finish off on an even gloomier note, we are facing an insect apocalypse. Across the world, there is an 80% decline in insect numbers and insect diversity, whether it's Costa Rica or a national park in Germany. There is a loss of beetle insects in all departments. What is causing it? We don't know. Is it pesticides? Almost certainly. Is it habitat loss? Almost certainly. Is it artificial light? We've added that now to the recipe, and I think we've also added elevated CO2 to that recipe. So it's death by 1,000 cuts. That's what we're looking at. And I think we need to think about our interaction with these organisms and the effect that we are having on them. And this relationship that we've had with dung beetles is a deep, deep relationship that goes back 3,000 years or more to the Egyptians. And even though we don't revere them as a god anymore, as they did in the past, I think they have proven themselves to be an incredibly useful sentinel and model animal for studying all sorts of aspects of biology and our effect on the world around us. And just like to say thank you, and all my colleagues that have contributed to this over the years. Thank you.
Thanks very much, Marcus. Thanks again for a lovely presentation. Are there any questions for Marcus from the audience and online? I think you frightened them all, Marcus. I was reading something this morning about, I think Natural Geographic published something on insect deaths on windscreens. You know, they collected dead bugs as we travel through the freeways. I'm sure that with habitat destruction adds to another cut to the insects. I mean,
yeah, and I think, just to add to that, it's been described as a snowstorm of moths. And I don't know how old you are, but I'm old enough to remember that you drove anywhere in this country and you had to stop and clean the windscreen. Doesn't happen anymore. And so you have seen directly the decline in insect numbers. And I think we should all be worried about it. I haven't got a solution. Switch off the lights. That would be the first one.
Also the question, I don't know whether you see me.
Go on, Alex.
Yeah, thank you, Marcus, for the nice presentation. And my question: do you know whether this, whether there is a contribution of the modern anti-helminth drugs which are used by cattle to the extinction of the dung beetles, in particular ivermectins, which are blocking the molting, and it's targeted against the nematodes, and the contents of the ivermectins in the dung sometimes is high, so probably it's a combined influence of the light pollution and the ivermectins which kill the dung beetles.
Absolutely, yeah. There's been a lot of work on ivermectins in dung, and there've been conflicting. Interestingly, there's been conflicting results from Europe and from Africa, and overall, though, the effect is a negative effect. You know, it's like if we take any drug, there's always a side effect, and if you're controlling worms in your cattle, you're going to affect the insects that are breeding in that dung. And you might see it as beneficial to suppress the flies, but you're also going to suppress the dung beetles. And the flies have a shorter life cycle, so they'll become resistant to the pesticide much quicker than the beetles will. And you get onto what's called a pesticide treadmill, that you continually use the pesticide to kill the pest, and when we should be using parasitoids like your little wasps, they're superb biocontrol agents. But yes, ivermectins are bad.
Thank you.
Thanks.
There is addition to that, and that is that one of the inhibitors of wind signaling is known previously as one of the anti-helminth sort of medicines. So it is possible that the effect of such medicine used even in, let's say, animal maintenance or in agriculture will be much more profound and affect evolutionary conserved signaling pathways which regulate cell division, etc.
Thank you. So you're giving us a scientific explanation for why they affect the development of the animals.
Well, it's one possible explanation. I don't think anybody really seriously look into that issue, but that's just a thought that I happen to know that such inhibitors are being used for, let's say, repurposing of existing drugs, etc.
Yeah. Thank you.
Any questions for the audience?
Thanks very much.
Thanks, Marcus. Thanks again.
Thank you. Thanks, Neville.
Our last two speakers, or last speakers for the session, Dr. Alexander Zinenko. No, sorry, wrong person. Dr. Andrei Fredchuk and Dr. Eugene Dickey. And they are from the National Antarctic Scientific Center of Ukraine. And they're going to talk about Antarctic research, a prospective field of cooperation between Ukraine and South Africa. Thank you. Over to one of you who's going to be presenting now.
Thank you very much, dear colleagues. Greetings from Kiev, from National Antarctic Scientific Center of Ukraine. Unfortunately, main presenter, our director, Dr. Evgen Dickey, is right now attending to another meeting. So I do not have a special presentation for this meeting, but let me say just a couple of words about Antarctica. It's a good point for cooperation. As you may know, Antarctica is one of the best planetary nature laboratory to research different parameters of environment, from tectonosphere to geospace, or biosphere and marine environmental. And what is more important, that the Ukrainian Antarctic Station and South African Antarctic Station located in different part of Antarctica. And it's very important and useful to compare monitoring data to develop forecasting models. And as you know, since full-scale Russian military aggressions in 2022, Ukrainian ice-class research vessel Noosfera, now based in Cape Town, and we're very grateful to South Africa for this opportunity to serve our vessel, because home port Odessa, it's not possible to return to Black Sea while this terrible war is going. So that's why we, while this ship situated in Cape Town and make regular research voyage to Antarctic Peninsula, so we can also make our research area more bigger and cover, for example, Weddell Sea or other part of South Ocean. So from this point of view, it's very important for us. Unfortunately, our bilateral cooperation in Antarctic field is not active yet, but we have enough possibility, good potential to do it, because both Ukraine and South Africa are a treaty member of Antarctic Treaty system, and we have Antarctic stations, year-round Antarctic stations, and we have also research fleet. So our joint efforts in field camp, in logistic operations, in measurements, it will be very, very effective and useful for our Antarctic programs. So that is my message to you, and let's think about it and make our cooperation more proactive in future.
Thank you, Dr. Fedchuk. So I do hope that the interaction between our own Antarctic program, which I know is quite active, and the Ukrainian one does proceed. I know it's difficult at this time, and we wish you all the best in terms of the war and in terms of your future research in the Antarctic. Thank you very much for that statement.
Thank you again.
We do have a few minutes before tea, and I did pick up a question online for Alex on your parasitoid wasps, Alex. It was posted on the chat in Zoom. I don't know if you can see it. I think it was from Natalia.
The questions I replied there. Yeah, there was a question whether what is the reason of the why emerged parasitoids reject the host, and I said I have no idea. I tried many approaches to block it, and I fed them with different media and also with nectar of flowering plants. So I presume they need something in what they get in nature and what they don't get in the laboratory. But what it is, I tried to provide them with everything what I presume they need, and it didn't help. So yeah, it's one of the intrigues to be solved sometimes, somehow.
Thanks, Alex. There was another question, not for you, but for Marcus Byrne. Marcus, I think it was from Natalia again, and her question was, and very intriguingly, because they don't understand that we have load shedding, they talked about the blackouts in Ukraine, and the same would be applying to us in terms of load shedding. And she asked mainly, will that aid in terms of dung beetle movement and orientation, I suppose?
Thanks. The short answer is no, because in reality, obviously, dung beetles don't really occur in an urban environment, or that many species do. But as a representative of many other animals that do, I think it will help. And in fact, one of my colleagues sitting behind me could probably answer that question better, because Siobhan is actually investigating the effects of our load shedding, our blackouts on the bat community. And what's the story so far?
Sure. Use this. Okay, thrown under the bus. Thanks, Marcus. No, so about a year ago, a colleague of mine approached me with that exact question of, can we sort of leverage load shedding to look at the effects of artificial light on animals in an experimental way. And so I said, yeah, let me find you some data. And I went and tracked down our sort of load shedding schedules or stages from 2014 to current. And then we also looked at some satellite data, so artificial light at night across the city of Johannesburg. We're actually able to show that there's a significant decline in light pollution as load shedding has been implemented and increased. So it declines by about 50%. So I'm going to probably botch the measures here, but it goes from about 400 radiance levels to about 200. So there's actually a marked decline in light pollution, and we think that there could be an effect on sort of short-term behaviors in some animals using urban environments. So we've got some money to put trackers on owls. So we're looking to see how artificial light at night or the reduction of that light pollution can affect foraging success in urban environments. We've got bat detectors going out across the city of Johannesburg, so looking at how that could affect bat community success in terms of foraging or activity, and it could go either way. So some of them will be drawn to light and others will be affected by light. So the responses are quite complex. And then I'd like to do something on rodents as well at some point. So I think there's some interesting stuff we can do in terms of giving up densities, sort of risk in terms of how far they're happy to venture out from bushes in more dark environments, I guess. And so that's kind of linked to those sort of owls and that foraging. So yeah, it's a good question.
Thank you. I think this is very much a collaborative team effort, as we can see. But I'd like to thank the three speakers for their presentation this morning. Very intriguing in many different levels. And we'll recess now for a tea break, and I'd like you back here at 5 minutes to 11. Thank you very much, and thanks to the speakers this morning. put it here. Okay. You can also look at it there. No, this will project. No, not the second slide. Welcome back, everyone. It's my pleasure to introduce Dr. Hanli Engelbrecht, who's also a member of our staff here at WITS and the School of Apes. And Hanli will be talking about species delimitation and biogeography of African green and bush snakes. Over to you, Hanli.
Good morning, everyone, and thank you for having me. So, as mentioned, I'll be talking about these beautiful snakes, the African green and bush snakes in the genus Phyllothomnus. Jumping straight in. This is a genus that's relatively speciose. It's currently sitting at a minimum of 20 species and counting. As a whole, the genus have a sub-Saharan distribution, but some of the species are specialists, habitat specialists, so some of them are either forest or savanna specialists, while others are habitat generalists. And as you can imagine, the extent of the distributions vary significantly. Indicated here with the orange polygons are just some of the species distributions depicted. One thing to show you here just how restricted some of the forest species are. And my main goal with this particular study, and it's a series of studies. The first question we wanted to know is, what are the evolutionary relationships of these species, given the widespread distribution? And at this particular point, when we're considering the species, they were only described based on their morphology. And so prior to this work, no one has yet undertook a phylogenetic revision. And this is exactly what we did in 2019. And we quickly learned that the taxonomy of the genus, when we compare morphology and phylogenetics, are not quite matching up. And so here we see what we found was, and so besides doing the phylogenetic studies, we also wanted to test, I forgot to mention now, just how valid some of the status of some species were. And so here, just a few examples, we saw that already some species at this particular point that were recognized as subspecies should be elevated to full species. Some species here in red, we have the spotted bush snake, turn out to be multiple lineages valid at the species level. And what was quite interesting about this particular species here is that those lineages did not even share a common ancestor. There's some other interesting cases, and just last year we described the second species that we discovered for Carinatus, the Carinatus 1, currently named after colleague Phyllothomnus chifunderi. And given all of these dynamics from a systematic point of view, the next question was, now that we know what the evolutionary relationships are, and now that we know it doesn't quite match the morphology, what is driving this diversity in the genus that is not reflected at the morphological on the morphological platform? And so the next question was then, and so here I just quickly want to recap, compared to the systematics prior to 2019, we see here everything that's highlighted in gray is what was consistent with the morphological species. And the majority here in green are species highlighted that were inconsistent with the morphological systematics at that particular point. And so these are mostly cryptic species. Very interestingly, we also find that two species in West Africa should be regarded as a single species, that Gerard is actually not a valid species. And so the next thing, as mentioned, was now to understand what is driving these relationships amongst these species and the speciation events in the genus. And before I get to that, I just want to remind everyone what the habitat classification looks like for Africa. Okay? And so this is the Copenhagen map, and we see that roughly if we look at the, especially here, the forest-restricted species, they seem to sort of correlate with our vegetation and climatic regions of the continent. What is not shown here is the savanna specialist and the habitat generalists. And the next question was now, given the distributions that we see, we have forest specialists, we have savanna specialists, we have widespread habitat generalists. Is a likely driver behind the evolutionary relationships of these species, could it be the forest-savanna dynamics, right? And a lot of you would be familiar with this, but I'm just reminding everyone else. And so what I mean with the forest and savanna dynamics is we need to remember that about 50 million years ago, Africa was nearly a pan-African forest, right? And about 20 million years ago, because of global decline in temperatures, more seasonal patterns for rainfall, the landscape became a little bit more heterogeneous. So we started to see the emergence of savanna. And later, in late Miocene to present, we started seeing savanna actually dominating the landscape. And we also started to see the emergence of grass systems. And those dynamics, the forest contracting, savanna expanding, starting to dominate the African landscape, how that impacted the evolution of African animals, specifically the megafauna, is quite well documented. Okay? And what we see for... Sorry. What we So what we see for the African megafauna, we see that in the late Miocene to sort of late Pleistocene, we see that there was an increase in the number of genera, right? And that can mainly be the main reason for that is it's because it's in that particular period, late Miocene to late Pleistocene, that we started seeing the savanna and grass systems dominating the landscape of the continent. And you can now connect the dots. How that compares to African reptiles, slightly different. We see that for the African reptiles, the Miocene period played a more significant role in as a driver for lineage diversification for reptiles, and the likely reason for that is the general life histories, right? The respective distributions of reptiles with respect to, or in contrast to your African megafauna. And what is different during the Miocene is we had these concomitant expansion and contraction of the forest and the savanna habitats. So it was cyclic events, right? So if you had forest expansion, you'd have savanna contraction and the other way around. And so given all of that, knowing how Phylothalmus is distributed over the landscape, I thought, wouldn't it be a cool question to specifically check out if it is the forest and savanna that drove lineage diversification within the genus? And I thought if we go about it, we need to be very specific. We need to have very specific hypotheses to be absolutely sure, because you know how biogeographers get usually picked on for not having specific hypotheses and just finding patterns and making sense of the patterns. And so I thought it would be a good idea from the get-go to have some hypotheses. And so what I specifically asked is, given the history of the forest, and specifically the Pan-African forest, when it was still, given the history rather of the forest and savanna biomes, it probably makes sense for forest specialists within Phyllothalmus to be paleoendemics, right? And to be the ancestor of the savanna taxa. And in a bigger scheme of things, I also thought that this would contribute to general herpetology, right? General African herpetology, because we do see many other snakes with similar distributions having within specific genera, having forest specialists, savanna specialists, or a widespread generalist. And in particular, those that's closely related to Phyllothalmus. And so this work would then also help us to understand what potential drivers in general could be for snakes, other snakes with similar distributions. And so, of course, to answer that question, we need to make sure, or we needed to know, did these snakes live through those events, right? And so Phyllothalmus belongs to, and many of the other snakes that I showed now, belongs to the family Colubridae. And what do you know? The Colubridae diverged from their sister families at about 50 million years ago. So we're good to go at this point. And so to answer the next part of this, the next bit of questions, so the storyline, what we did was we took the existing phylogeny and we dated the phylogeny. How we went about it was choosing closely related fossils for which we know the dates of, and also dated by geographic events. And to spice things up a little bit more, we compared Phyllothalmus with a second genus having similar habitat, species with similar habitat distributions and affiliations. And so besides dating the phylogeny, the next thing now we needed to do was reconstruct the habitat associations for the species over evolutionary time, right? So we know now which habitats they're affiliated with, but we needed to understand what did that picture look like if we traveled back in time. And so what I did was went for the simplest route. We know that there's many subclasses of forest and many subclasses of savanna, and I went, let's group them all together and classify it as closed habitat versus open habitat. And that also then allowed us to include other biomes such as your fynbos and the arid, semi-arid biomes into the open habitat category, right? And so why I'm saying that is because there are a few species that are associated here with the fynbos and the arid, semi-arid biomes. And so what we found was that Phyllothalmus as a genus emerged in the early Miocene, right? At about 20 million years ago. And indeed, we found that the ancestral state for these, the genus, habitat state, was forest. So the genus emerged in forest. We then see that there's, for the two main clades, we see that most of the forest specialists sits in this one particular clade, and where the second clade is is a little bit more diverse, right? And so we had these two clades split in the early Miocene, and so actually to streamline the story line, I'm just going to say what it is important to look at here and to note here is that every time that we see that there was a speciation event and transition into different states, it was associated with main changes in the landscape, okay? And so what we see is that some of the species, all of the forest specialists, maintained the ancestral state, except for Natalensis, which recently went into forest. And we saw that each time we had a savanna expansion event, we saw that species started transitioning into open habitat. So much so that some species actually specialized into open habitat, which personally, knowing the genus, I find very interesting. It is quite odd for some of these species to be specialists in open habitat, specifically in southwestern Africa, in this arid, semi-arid areas. And if we compare that to remember I said we included, just for interest's sake, another genus, we wanted to compare what that would look like. We see that for the African herald snakes, emerged at a similar time as Phallothamnus. However, the ancestral habitat state here is they were generalist. The ancestors were not a forest specialist, but instead they were habitat generalist. And what is probably the most obvious thing that you would have noticed is two genera emerging at the same time. One is relatively more speciose than the other one. Okay? And we also see that compared to Phallothamnus, we saw that forest specialists, as we know them recently or today, moved into forests recently, right? And one of my first questions was, if these species emerged at the same time, lived in the same spaces, experienced the same events, why are they showing these different patterns, right? And two short answers. First of all, as mentioned, African herald snakes were generalist. So they would not have been as impacted as the forest specialist having to live through these habitat alterations. And probably more important, the African herald snakes are terrestrial, whereas Phallothamnus are primarily arboreal. And so being highly dependent on trees, you can now imagine how that would have led to, how those events would have led to more speciation events. Phallothamnus, towards the end, Phallothamnus, given these results, it is now very interesting to take it back to other widespread African snakes occurring in similar habitats. And the take-home message here is that although species share similar distribution patterns, and they emerge at the same time, and they all lived through the same events, the results, the speciation results, can look very different, and we've started to see that is the case for other genera, can look very different, and what one should remember is to consider the different life history traits of the different genera that you're studying. And the next bit of work that we're currently doing is looking at whether these radiations are adaptive. And Phallothamnus has been a foundational species, or foundational genus in addressing adaptive radiation species with respect to these historical events for many other snakes. And besides this, the systematics, that's going to keep me busy for a very long time. That is what we're currently addressing in our lab. And so I'm hoping to share these results with you soon. We're starting to see that there is actually some climbing and feeding adaptations associated with this radiation. On that note, I'd like to thank you for listening and ready to take any questions.
Thanks very much, Hanli. Any questions from the audience? I see Sally.
There is a question online
Hi.
from Dr. Janelle.
That was really interesting. Thanks, Hanli. I suppose it's more of an ecological question. I was wondering what it means to be a habitat generalist if you're a snake. are there really species that are found in forest and grassland environments today? And you think those ones were actually around like also 10 million years ago?
No. Thanks, Sally. So to answer the first part of your question, yes. You'd find those same species in forested or savanna habitats. And no, they would not have... are you now referring to the herald snakes, I'm assuming, when you ask? And so what is interesting with the herald snakes is that if we take time into consideration and we look at those early species that were habitat generalists, you look at the specific distributions, it correlates to areas, specifically in East Africa, Ethiopia, and so on, where savanna emerged for the very first time. So in that regard, it adds up.
There's a question online from Alexander.
Hello.
I will check if you can hear me, because I'm next presenter. It's crucial for me too. But the question is, what kind of data have you used for these phylogenetic reconstructions, and if any demographic reconstructions are possible to check any of your hypotheses based on this data, if it is genomic, for example?
I'm not sure if I follow the question, Alexander. So for the reconstruction, are you talking about the habitat reconstruction? The habitat affiliation?
No, phylogeny.
I'm not following. Is anyone else following and maybe want to clarify?
What kind of genetic data?
Oh, genes. Are you referring to genes? So sorry. So we used a combination of 5 genes, and so it's a combination of 2 nucleogenes and 3 mitochondrial genes, all at with different mutation rates, as to not be biased. And so specifically it was the CMOS gene, RAG1, site B, ND4, and 16S ribosomal. Does that answer your question?
And to follow up, is it possible to do any demographic reconstructions based on these sequences? Maybe they can inform you about the past of this species.
Honestly, that is I don't know. I'm not too sure what you're asking. Are you referring to looking at how population numbers would have changed over time? Is that what you're asking? So population density. I'm sure that's possible, slightly outside of my research expertise, but totally possible. Our markers would need to be different.
Thank you.
Thank you.
There's a question here.
So with regards to the herald snakes that were generalists and then eventually became specialists, do you see any morphological characteristics that were retained that would work perfectly for an open habitat, although the snake is in the closed habitat?
Are you asking, are we seeing ecological or ecomorphological traits that link them to habitat specialization versus generalization? So at the moment, feeding, yes. And so what we see is, and Naughty Kutlana for asking this question, what we're seeing, so the herald snakes are forest specialists, and what we're seeing is that it's literally a case of, it seems to be a case of feeding specialization. And so they're taking frogs that have similar distribution patterns and share similar life history traits. At the moment, that's what I know of, that feeding specialization. My priorities lies elsewhere with phylothalmus at the moment. Yes, Sally.
If there's time. You started off your talk talking around how with large mammals they associate diversity, increased diversity with more open habitat. And then I think you argued that when you're looking at snakes, what's interesting is more the kind of transitions from forest to grassland. So then just generally, do you think diversity, I've always had a problem with the idea that there's more species of megaherbivore, therefore there's more open habitat, because diversity, like your argument about the snakes makes a lot more sense, which is that if you have these transitions, you expect diversification events. So do you think the processes that created diversity in snakes are really different from the megaherbivores? And is it okay to associate diversity with like extent of habitat? That seems to me a bit sort of strange thinking.
I totally agree with you, Sally, and I think that was one of what was one of the take-home messages for me with this particular study, specifically if you look at the two genera of snakes that we compare. Yes, habitat alterations seem to have, to a large extent, in a very generalized pattern, have influenced lineage diversification and speciation events. But we cannot forget to account for the different life history traits of the species. And so it really depends on the ecological preferences, is a very not nice word to use, but the ecological habitus, right? And so there's a classic example from the herald snakes and the green snakes, that given that they have different ecological habitus, they lived through the same events, but were impacted in different ways. With regards to the African megafauna, what I meant there was, and it seems like at the beginning it could be something like, I almost want to say ecological release, and it depends on their life history traits, right? And so it is because, my argument is, it is because of their life history traits that as savanna expanded, we could start accommodate for more species specifically associated with savanna habitat types. So yeah, it's not just two different mechanisms. I'd say it is a mechanism depending on the life history traits of the animals that we're studying, but either way, given where they find themselves, they're going to be impacted by these habitat alterations, and depending on their life history traits and the ecological habitus, some might be more severely impacted than others.
I've got one question before I let you go. You talked about the emerald snakes and the other ones. Is there any, you said from the same habitat, do they occur together and there's any chance of interspecific competition between them that drove any of these processes as a hypothesis?
The herald snakes compared to the green snakes. Yes, they occur together. And I'm going to slightly shape your question a little bit. I think, so competition from an ecological guild, for sure, because phylothalmas are to a large extent, they also feed on frogs. What is more interesting is, I wouldn't go as far as comparing the herald snakes and the African green and bush snakes directly, because they they're very distantly related. What is more interesting is if we look at the Dysphalidine tribe, so it includes the boomslang, the vine snakes, and the African green and bush snakes. So they're more closely related. And I'd say that is where the competition is happening, and something that's fascinating me at the very moment is exactly that, is how those genera are competing, and you see it in their climbing proficiencies, differences in their climbing proficiencies, but also specialization and generalization in terms of feeding. So there's niche partitioning yet to be tested. But definitely we see it when it comes to the feeding adaptations, and you'll see that the boomslang, for instance, is way more of a feeding generalist than what Phylothalmus is. So Phylothalmus is frogs and lizards, and that's about it. Boomslang, we have frogs, lizards, small birds, and other reptiles. And then sort of a similar situation for the vine snakes. And so you can actually rank it depending on who came first in the tribe. And it seems that Phylothalmus just exploited the frog lizard feeding niche.
Thanks very much. And I think we need to give only a round of applause for a presentation. Thank you.
Thanks again. Thanks again, Hanli. We're moving on now. I don't know if you can hear me through the mic. Okay. To Dr. Alexander Zinenko, who actually asked a question of Hanli, so he's continuing with the theme. And Alexander is in the Karazhan National University in Ukraine. And his talk is on Ukrainian contributions to research on African amphibians and reptiles. Over to you and welcome, Alexander.
Thank you very much. Unfortunately, I'm not in university now, not in my office. You know that the situation in eastern Ukraine is specifically very severe, and I'm now in western Ukraine, locally translocated, working from my home online mainly, and that explains my background. When I was preparing my presentation, I was thinking more about, my personal dimension of African research, and also what Ukrainians are doing in Africa and why. Yeah? And the situation with collaboration between Africa and Ukraine is such that we don't have much contacts. We're far away, and Ukraine doesn't have much resources to do tropical biology and description of it, biodiversity elsewhere, except of territory of Ukraine and some other places which are not numerous. And I will share my presentation now. Just a second. It's live. And and if to speak about history, Ukrainian herpetologists came to African fauna and Africa as a continent mainly virtually, and our objects lead us to this wonderful continent. And a good example is Mykola Sherbak, who was a renowned taxonomist who did a large revisions in arid species of reptiles, worked mainly with the collections in European museums, but also remotely. He was not even able to leave Ukraine for a short time until the collapse of the Soviet Union, because in his young years he was accused of Ukrainian nationalism and sent to Gulag camps in northern Russia for 5 years, and after that managed to keep on a track and receive an education, and did a lot of research in the field of herpetology, but until the end of the Soviet Union was never let outside So he tried to exploit the vast territory of the Soviet Union and became a traveler and researcher and zoologist and taxonomist within large enough country to fulfill his scientific interest. And among the groups he was interested in were geckos and lacertids, which were present also in African fauna. And to complete taxonomic revision of a large group, he had to go to the collections and to the taxa present in African fauna. So that's the typical way of how Ukrainians go to Africa, with a few exceptions. And all this situation, similar story with another employee of our Institute of Zoology, Schmalgaus Institute of Zoology, who is a co-organizer of this memorial conference. Anatoly Tokar worked with erics, and since some erics are present in Africa, he also did taxonomic revisions of these species and worked with a collection. Otherwise, Africa for Ukrainian herpetologists is like a heaven, yeah? And not only because of beautiful and interesting and diverse fauna and landscapes and nature in general. Many questions, not only in taxonomy, but in evolutionary biology. Africa is like a heaven for Ukrainian herpetologists because those who were able to go there rarely coming back, and we know about it mostly from the books and from the scientific publications. In my case, I was lucky to go and to come back. And actually, I was and worked in the field in Africa only once in my life. I'm giving a personal dimension to my presentation. I'm sorry, I was oriented into this format of contribution. It was probably some kind of my mistake. But anyway, I have been in Ethiopia one time in 2018, thanks to National Geographic Explorer grant. And we are the team of international scientists went there to explore and to describe primarily biodiversity of a Harina forest, which is one of the last large forests in Ethiopia, where population is growing fast and natural habitats is declining. And the organizer of this expedition is Artur Tutenko, a zoologist, and he is a Ukrainian expat, but he refused to call himself Ukrainian zoologist because he is based now in Bavaria in Germany for at least 25 years. And he has no Ukrainian citizenship or affiliation and probably consider himself as a German. This is also, I forgive my friend Artur Tutenko that I'm describing him, but he is also a typical Ukrainian in that respect, being not able to continue what he likes, and career in zoology was his dream. He moved to another country, probably the same way as Boris Ballinsky did, and as for Artur, he would never achieve what he achieved in Ukraine, and the same probably applies to many of us. And when in Harina forest, we were just amazed naturalist in a new place. Of course, it was interesting experience, and I personally personally didn't expect many things which I saw in Ethiopia, both good and bad. But first we just do observation of local fauna and try to describe it. So we were much behind the time and on a descriptional stage of biology, zoology. Still, we found that there is a place for contribution even in such simple activity, because many of species are either not completely described, or some biology of these species are not known or not described properly. So first we made the pictures, and later described a tadpole of Leptopelis ragazzi. It's funny tree frog which lives in a forest and reproduce in the streams below the trees. Our main goal was to find and to understand the distribution of large Betis harena vipera species. We did succeed. Instead of that, we found unexpectedly small frog on a glade in a small puddles, reproducing during the end of a wet season. And the name of this frog was Phrynobatrachus inexpectatus, purely, correct name for this species, for us, for our experience. And next observation of its breeding biology was mind-blowing for me, because you have a huge glade with many petals, and only few frogs collect in one small petal among the multitude. And it already became a thought-provoking, and in addition to description of reproduction cycle and development of these species in natural conditions, we did this in a comparatively short time. This species goes from the egg to developed pre-metamorph tadpole within few weeks. Much faster than we used to see in European frogs and toads. We also saw that there is a different conditions in which local fauna lives, and these conditions are drastically different from European, where we were trained and had the most experience of a field zoology. This aggregation in this one small petal probably dilute the possible pressure of predators, and even looking under your feet and walking on a forest in Ethiopia was interesting for outside zoologists. And our descriptional stage of exploring of African fauna continued, and taxonomy of African species became a nightmare for us. And after trying to find out what species is what, we came to the conclusion that not only we don't know, but many species are lacking proper description, and type collections collected during 19th century were not collected in a proper way. Locality was not indicated in the way that you can identify the place now. Study of the group research of molecular phylogeny of this group may lead to the conclusion that there is not only one but many species, but since the type territory was not indicated properly, you cannot identify what species was originally described. And this is true for many groups of African frogs, toads, snakes, and lizards, but also this specific example corresponds to Leptopelis genus of tree frogs. And to disentangle this riddle, we had to go to collections and work with original type specimens, which are discolored, dismorphed, and poorly preserved because of time, of course. We had to read original diaries of collectors. These decipher old names of localities, put them on a current map, align with the results of phylogeographic and phylogenomic studies, add morphological description and description of life stages, record their voices, their calls, and altogether will bring taxonomy, I believe, not by only our efforts, but by the common efforts of different researchers to it will bring taxonomy of the group to the stable and better condition than it was before. And this was my contribution to the region herpetology, and I consider this as my side project, but still I am glad to take part in this. And then after descriptional study, when you finally understand what you're holding in your hands, you see the evolutionary story unraveling in front of you. Again, to emphasize the difference between European herpetofauna and African herpetofauna, I must say that European herpetofauna was mainly affected by climate fluctuations and glaciation cycles, which made most of the territory of Europe uninhabitable by reptiles and amphibians. And what current species of amphibians and reptiles do in Europe is running from refugia to the north, and then probably back to the refugia after the next cycle. And there is a much fewer species and much less ecological speciation between species of reptiles and amphibians in Europe. Here in Africa, in a more stable climatic conditions, they are changing, but not that drastically. You have a chance to see very different, amazing evolutionary stories unraveling. And this tree frog I showed you before lives on the trees, and it belongs to the same genus as other species who live on a altitude up to 3,000 meters. Leptopelis montanus, renamed by us, is an example. And instead of climbing on the trees, it lives in a wet grassland and have to hide in the soil and bury in the soil. And it does it in the same way many burying frogs does, using structures on their hind feet, and within few million years. In the same genus, several species diversify in totally different direction and have specialization, one living on the trees with a typical morphology of a tree frog, and another drifting towards burying frog, which, for my point of view, is something completely different. And my original study object in my PhD studies and later on a postdoc were vipers, small vipers which live in a temperate zone of North Asia and Europe. And I was trained as a taxonomist and evolutionary biologist in this group. And in case of my Ethiopian experience, we found several snake species. And again, trying to identify them lead us to understand that there is a lot to be done in this group too. Snake, which was previously called Lamprophis erlangeri, after sequencing few standard genes on which reconstruction of phylogeny of African snakes are usually done, revealed that this species has a genetic distance equivalent or similar to genetic distance between different genera inside Lamprophid snakes. And after a careful description of internal anatomy and external morphology, we described the species or elevated it to the new genus Bopha. Together with Arthur. And again, to receive a context, not to put this new genus in a space, we have to analyze both phylogeny based on the same genes and morphology of a closely related species of Boodantini. And it was amazing to me to see how much difference accumulated in phylogeny of African snakes. And you see large diversification both in body shape, size, head especially has different morphology and different structures, and lacking of knowledge about biology of these snakes prevent us from understanding what are these structures for. Pupil size and shape, eye size and shape, and other morphological features are so different that it just makes me believe that still it is a lot to be done here too. And I am from a group, I remind you, where two species look, as for taxonomists who probably work with African snakes, you will say that these are the same. So on the left side is a steppe viper, on the right side is a Kaznakovi viper, and they are considered to be a very good species. And within each group, there are from 7 to 10 new species described recently. In spite they are hard to recognize. Even the maximal difference between them is so small that you can mix them up. And even in the middle, the hybrid was described as a species. And the age of differentiation of this group spans from 5 to 8 million years, almost nothing compared to African, to tropical species of snakes. And speaking about the differentiation and specialization to different ecological niches, snakes are quite poor in characters of external morphology, but if you look inside the skull, you see the great differences. This is just an example, and we continue our work and try to employ CT scanning to provide morphological description based on external morphology with internal morphology, and it is much more informative, but much more difficult to formalize, and we are probably struggling with formalizing the characters of osteological characters. And at the end, something which corresponds to the previous contribution, which I found very interesting and fascinating, is a simple idea of putting on the same page of our publication with redescription of Boffa, with description of a Boffa genus. We put on the same page, on the same illustration, ranges and simplified phylogenetic tree of genera of Boadentina snakes. And we already on this stage, without any formal analysis, can formulate the main hypothesis that ground-dwelling forest species of Boadentina probably originate from East Africa, upland, probably from territory of a mountain forest of Ethiopia or near that. And the first split between Pseudoboadon and apparently relict species of genus Boffa remained there. And then dispersion and speciation events lead to emergence of Botrolicus and Botrophthalmus, which moved a little bit to the west, to the tropical West African and Central African forests. And the next stage of evolution of this group was transition to savanna, to open landscapes in Boaidon, and conquer of the territory of Africa, and even going outside of Africa to Arabian Peninsula, and move toward the north, toward the South Africa of three genera of Lycodonomorphus, which indicate the route that the group used to this dispersion along the Rift and Great African lakes, and finally Lamprophis and Alopesion rested in South African landscapes, territory. Even if I will not continue this work and something prevent me from that, I am very thankful to the Artur Tutenko for introducing to me African research. I hope that some kind of collaboration will continue, and it will not be only taxonomic studies. I'm very grateful to organizers of this conference to giving me the floor to speak about such interesting and fascinating processes. And thank you very much for your time. I'm open to the questions if there are any.
Thanks very much, Alexander. Are there any questions from the audience?
Thanks for that, Alexander. I've got a question about Leptopilus ragazii. Does it make a foam nest? Because it looks very like our Chiromantis cerampolina that does the same.
Excuse me, can you repeat? Leptopilus ragazii, does it look like what? I didn't hear.
Does it make a foam nest in the trees above the water?
Probably not. It's not known for this genus in general, and they reproduce just in streams. And perhaps this adaptation of making foam nests is adaptation against predators in the ponds where frogs, I don't remember, Rhacophorus probably lives. In South Asia. In the case of Ethiopian Leptopelis tree frogs, they breed in water bodies are lacking predators, and I think there is no sense to make a nest on a tree because there is no benefit from it.
Thank you. Any more questions? Alexander, thank you very much for your presentation. I do hope that you can continue your research, as you pointed out in your last slide, and you're welcome to visit us in South Africa to continue some of that work if you needed to. But the very best to you, and thanks very much for your presentation. Thank you.
Thank you.
Do you need some water?
No, I'm great.
Okay. So we got about lunches at 20 past. So
okay.
Yeah.
That's
so
great.
Go as long as you need.
Okay, thanks.
Do you need to control the
I'll just
slide?
show them which file it is and which time.
Anyway, thanks again for coming.
No, it's a pleasure. Thank you. It was a lovely meeting. Yes, it's too many. Yes, it's fun. There we go. Thank you all.
Do you want to control it from here?
I can do that.
Yeah. Good. It's now another pleasure to introduce Professor Jennifer Botha. She's the director of GENUS, and some of you might know what GENUS is. It's associated with the Evolutionary Studies Institute, and it's the Center of Excellence in Paleo Sciences. So it's a warm welcome to Jennifer. And she's going to be speaking about past mass extinctions and how they can help today's crisis. Thank you very much. Over to you.
Right, good afternoon, everybody. Just want to see how this works. Has the slide changed? Yes. All right. Hi, so I'm Jennifer, and yes, I head up a Center of Excellence in Paleosciences called GENUS, but I'm also a researcher in the Evolutionary Studies Institute. And my interests are in paleobiology and paleoecology, but I focus on differential species survival during dramatic environmental change. So I study past mass extinctions. And for some 20-odd years now, I've been focusing on the end-Permian mass extinction, or the Perma-Triassic mass extinction, which occurred some 252 million years ago. And it is fondly nicknamed the mother of all mass extinctions because it changed the global community structure of both terrestrial and marine communities forever. They were never the same again. And it's considered to be one of the most catastrophic biotic crises in Phanerozoic history. So, why study past mass extinctions? Well, when we look at these events, we want to find out which taxa or species go extinct and why they did so, what they were sensitive to, what they couldn't handle. We particularly want to know what the survivors were doing, what kind of adaptive strategies did they employ to be able to survive dramatic environmental change. And also, interestingly, some lineages actually take advantage of these events and radiate and become very diverse or very abundant. They are actually successful because of the change in climate. We want to know which taxa are doing that. And as an overall whole, we want to know how long it takes for ecosystems to recover from such events. And of course, this information can be used in today's world because, as we all know, we're currently going through the 6th major mass extinction. This image here with each symbol of animal represents 100 species. So you can see that amongst birds, there's about 13% in red. In the mammals, 25%, and in amphibians, as much as 41% of these species are currently threatened. A study was done recently last year by, I think his name was Finn, when he looked at various clades, various groups of animals, and they looked at what they thought the 6th mass extinction might turn into, and unfortunately they are predicting that it's going to be worse than previously thought. When looking at amphibians, birds, and mammals, if you have a look at the red bars there, they are going to be the ones mostly affected. Reptiles, fish, and insects may be less affected, but if you have a look at those grey bars, that represents all of the unknown data. So we really still don't have a complete picture of what lies ahead. So as we all know today, greenhouse gases are causing an increase in global temperature, the main drivers being carbon dioxide, which is shown in red here, and methane, which is shown in yellow. And most greenhouse gases cause a warming effect. So our common, usually known gases, including carbon dioxide, methane, etc., cause an increase in temperature. But we also have aerosols from pollution that may even cause a cooling effect. So the big take-home message here is that there is an increase in variability. And I'm going to say variability and unpredictability quite a lot during this talk because those really are the key words to what we need to examine. So if you have a look at this graph here, you can see that basically since the Industrial Revolution, humans are adding towards the increase in global temperature. If you have a look at the green line, those are natural drivers only. But if you add human influence, you can see that the red line goes up quite significantly. But this is just in today's world. The Industrial Revolution, in terms of geological history, is actually not even a blip on our radar. It is so it's not even really considered the past. So what has happened in the past geological record? What has happened in Earth's history? Have we seen similar situations on our planet before? And yes, indeed, we have. If you have a look at the red parts on the graph, these are times when the global temperature was higher. So we have a lot of hothouse effects. We have global warming. And the blue areas are when life was cooler. When the environment was cooler, we have polar ice caps during those stages. And you can see right at the very end that we, in today's world, are still in that blue area. But just because other organisms may have survived these hothouse worlds doesn't mean to say it was a great place or a great time to live in. And you can see that our line is very quickly increasing towards that grey line. That blue line there is actually increasing at a very quick rate. So as I said, I study the Permo-Triassic mass extinction, and the reason why I bring up greenhouse gases and global warming and carbon emissions, etc., is because this is kind of similar to what happened to the most catastrophic biotech crisis in history. So although researchers have gone back and forth over decades, we now all agree that the main cause of the extinction was volcanic eruptions. There is a large igneous province in what is now Russia called the Siberian Traps. It covers about 2 million square kilometers in area, and they represent the largest volcanic eruptions in Earth's history. And there would have been explosions, pyroclastic flows, etc., but there also would have been times where lava just seeped out of the ground in a far more calm manner, except the only problem is they're still spewing out gases that go all of the way around the world. And this situation lasted for some 1 million years and is timed with the Permo-Triassic mass extinction. So how could volcanic eruptions cause such a global extinction? Well, obviously the lava and the pyroclastic flows would have affected the immediate area, and as I said, this area was about 2 million square kilometers, but that's still not really global in nature. But the sulfur and the ash would have blocked sunlight. The greenhouse gases such as carbon dioxide and methane and water vapor would have increased global temperatures by allowing the heat in, but not allowing it back out of the atmosphere. And there are also chlorine and fluorine gases in volcanic eruptions that destroy our ozone layer and allow harmful UV rays to penetrate, causing radiation problems. And some of these effects you can see last for hundreds and thousands of years. Other effects last for millions of years. So when we look at what the environment was like during this time, we can actually see that there were changes in climate. We look at the sedimentology and we can see that the environment changed from one of high sinuosity meandering rivers with expanding wet lowland floodplains through a straightening transition stage to one of braided ephemeral rivers. So these dried up every year, dry floodplains susceptible to flash floods. Although there were organisms that survived, it really wasn't a great place to live in at that time. So South Africa is very special in having what we call the Karoo Basin, and it includes the most complete, best preserved fossil record and geological record of this time period on land. So there are other sections around the world that preserve the marine extinction, but South Africa is number one on the list for visiting if you want to study the Permo-Triassic mass extinction in the terrestrial realm. Numerous studies over years have greatly improved the accuracy of our data, where we have used sedimentology, geochemistry, biostratigraphy to look at the biostratigraphic ranges, in other words, the times in which certain species lived from the uppermost Permian to the lowermost Triassic, and we can see the faunal turnover and what species went extinct and which survived and which appeared during the post-extinction recovery period. So with this data, we go out into the field and we collect fossils. And over many years, we have put together which taxa may have gone extinct, which disappeared during the extinction interval. I am mostly interested in the survivor taxa, so I tend to focus on those in the earliest Triassic because I want to know what kind of survival strategies they employed, what made them so special and so hardy that they managed to survive this really harsh, unpredictable environment after the extinction. One of the things I can tell you is that we find numerous burrows, fossilized burrows in the earliest Triassic of various different sizes relating to different species. So we have dicynodonts and cynodonts. These are therapsids, the ancient ancestors of mammals. And we have small burrows and large burrows, and sometimes we are lucky enough to find the fossils within the burrows themselves. And we do not think that this is a taphonomic bias. In other words, we don't think this is a difference in preservation between the latest Permian and the earliest Triassic, because the taphonomic styles are similar. So we really are seeing a true reflection of the environment in that most of these animals during the early Triassic were burrowing. So it really was some kind of strategy that was employed by a lot of animals during the early Triassic. They may have burrowed at night, they may have burrowed during the day to escape the heat, they may even have estivated or hibernated. We don't actually know why they were using these burrows, and this is something we're still looking at, but we do know that they were important for the survival of early Triassic taxa. So I'm really interested in looking at the biology of fossil animals. Now, how do you do that when it's just a piece of rock at this stage? Well, greatly enough for my life, is that fossil bone generally preserves very well, so that although all of the organic components have disintegrated, the positions of all the bone cells and the blood vessels and the tissue fibers, the positions of all of these remain intact. And so we can look at modern taxa, and we can look at the bone microstructure of these animals, and we can learn certain things from them that you cannot learn from traditional techniques like looking at the morphology of a fossil. And we can relate this back to the fossil record, to extinct taxa. So we can look at the growth rates, how fast or slow an animal lived or grew, the patterns, the way in which it grew. We can look at when they may have reached reproductive maturity, when they reached maximum size, how old the animal was when it died. We can even look at biomechanical adaptations. So for example, if you're looking at an amphibious animal such as that hippo there, or a burrowing badger, you can see that the bone microstructure inside is very different. So the bone reacts differently depending on the lifestyle. And we can also look at the effects of environmental perturbation. So those arrows, those thin arrows that I'm showing there on the blue and orange slide, this is a type of modern deer. Those represent annual growth marks. They're laid down once a year during the unfavorable growing season, usually during winter or the dry season. And you can actually count them and see how old the animal was when it died. So generally in paleontology, people will look at paleohistology and they will use this to describe the growth history of a particular species or of a particular specimen. But I was interested in using bone histology as a tool to expand and to use it as some kind of framework to ask the big picture questions. And I was interested in looking at whether life histories in vertebrates changed during the Permo-Triassic mass extinction. So I looked at one clade, one group of animals called therapsids. These are the ancient ancestors of mammals. And if you look at the blue taxa below the Permo-Triassic extinction interval, these are the Permian therapsids. And the ones on top in red are the ones from the earliest Triassic. Now, all I really want to point out with this slide is that you can see there are lots of arrows in the taxa in the purple area. And that just shows that they were actually growing for several years before they died. So what we see in the latest Permian are normal growth strategies where an animal takes several years to reach reproductive maturity and then grows for several more years after that before perhaps reaching maximum size or before dying. What we see in the earliest Triassic, though, is something completely different. You can see there are no arrows there. Either the taxa are very small and they take within 1 year to reach its maximum body size, or they are dying at very young ages. So they are not getting to a ripe old age like they were in the Permian. The life histories have indeed changed. And if we plot this using the biostratigraphic ranges and phylogeny, you can see the taxa in blue below the black line. These are Permian taxa, and these are taking many years to reach reproductive maturity and getting to a ripe old age, whereas those taxa in red in the earliest Triassic are dying at very young ages. So there is a very high juvenile mortality, and these animals are not living to a ripe old age. It's only until later in the middle Triassic do we see that blue kind of growth strategy come in again, where animals are taking their time to reach reproductive maturity again, and we start to see a Permian type of growth strategy come in. So I've taken some of the research that I've done on the Permo-Triassic mass extinction, and I've extended this. I'm now looking at the end Triassic mass extinction, which occurred about 201 million years ago. And when we look at the end Triassic, there are several groups of therapsids that went extinct, but there are also many groups of archosaurs. This is a type of reptile that went extinct, including phytosaurs. These are aquatic reptiles. We have many ornithosuchids, the etosaurs, rauisuchians. All of these taxa disappeared at the end of the Triassic. But you can see in red there, the crocodilomorphs, these are the ancestors of crocodilians, survived the extinction, and so did the dinosaurs. In fact, the dinosaurs don't seem to be negatively affected by the extinction at all. You can see that they're starting off in the late Triassic, but they go on, as we all know, to become very diverse and abundant and the dominant terrestrial animals during the Jurassic and the Cretaceous. And luckily enough, again, if I want to study the end Triassic extinction, the best place to be is South Africa. There are many places around the world that preserve this time period, but most of them do not have body fossils. They only have trace fossils, which are things like footprints and traces of animals that had lived at the time, but not the actual 3D fossils. So we don't get a full picture of what the ecosystem looked like at that time. But the Elliott Formation, which is latest Triassic in South Africa in the Karoo Basin, contains this event with numerous body fossils, new species that are being found every year. And the Elliott can be divided into two parts, the lower Elliott, and you can see that the taxa are different to those in the upper Elliott. And the end Triassic extinction is close to that boundary between the lower and the Elliott Formation. So when we look at the sedimentology, again, we see that there is a change in climate. In the lower Elliott, we see these meandering perennial rivers like we do in the Permian. And in the upper Elliott, we see these ephemeral rivers that were subject to flash floods and playa lakes, similar to what we see in the earliest Triassic. So again, we are seeing this dramatic environmental change. What we do have in the Elliott Formation is numerous sauropodomorph dinosaurs. These are the ancestors of those really, really large sauropod dinosaurs that got to about 70 tons in size. So there are a lot of sauropodomorphs in South America, but we also have a lot of these early sauropodomorph dinosaurs. And what's so special about these really giant sauropods? Well, how did they get to this giant size? So people looking at dinosaur biology have thought that, well, looking at their histology, they grow really, really quickly, and they also grow continuously. Their growth does not seem to slow down every year. So they think that these animals grew to such large sizes because they grew fast, and we see this in the true sauropods. So a colleague of mine in the ESI, he's the one going out doing the hard work and finding all these new species of dinosaurs, and then I bring them home and cut them up, so I'm often not very popular. But we really do get a lot of really important information that we would not otherwise get. And he and I decided to look at two sauropodomorph dinosaurs from the period of the end Triassic extinction in South Africa, Ardonyx and Cephapanosaurus, and we looked at the life histories of these animals. And what we actually found was that they were growing quicker than we actually thought. These are relatively small dinosaurs. By that I mean they are 1 to 2 tons in size, which sounds great, but compared to 70 tons, it's really quite small. You can see those taxa in blue are growing relatively slowly, but the Ardonyx and Cephapanosaurus dinosaurs, they are growing rapidly, although they had interrupted growth. So their growth slowed down every year, but the times that they were depositing bone tissue, it was really, really rapid. And similar to the Lissamsaurid dinosaurs, which they are closely related to, and those were about 10 tons in body weight. So when we look at Ardonyx and Cephapanosaurus, the 1 to 2 ton dinosaurs, which are relatively small, they were already growing quickly. And it seems that it's actually the other way around, that rapid growth was a prerequisite to allow the true sauropods to grow to such enormous sizes, that it was already there in the depths of the phylogenetic tree to enable them to reach these huge body sizes. I want to share with you another new species that's been recently found from the Elliott Formation. This is a crocodilomorph, an ancestor of crocodilians. He looks a lot like a crocodile. There's an outline there, but he was bipedal, so it's a little bit strange for us to imagine. We're used to our sprawling quadrupedal crocodilians. This animal, the tibia, is about 30 centimeters in length, so he was several meters in length. He was a large animal. And he's from the Elliott Formation from the latest Triassic. Now, as we know today, crocodilians grow quite slowly. They are typical ectothermic vertebrates, and they grow slowly, and they have interrupted growth every year. But we know from looking at the fossil record, if we look at archosauriforms, these are the ancestors of this group of animals, they actually grew quite quickly. So people think that crocodilians began growing slowly, that it was a secondarily evolved criterion for crocodilians, and that they evolved from faster growing ancestors. But we really don't know how and when this happened. So I looked at the bone histology of our new species of crocodilomorph, and I won't go into the details of the bone tissue patterns, just to show you that we see parallel fibered bone, which is a bone tissue that we find intermediate between the fast-growing tissues and the slow-growing tissues. There, the lines in green represent annual growth marks, so I can tell that this animal was at least 3 years old when it died. It was still growing as a sub-adult. But I wanted to show you that picture just to show you all of those holes in the bone tissue or where the blood vessels used to lie. And the pattern, the organization, is circumferential, and we call this a laminar vascular organization. Now, I just mentioned those two features because I want to point out to you in the phylogenetic tree of these animals, fast bone tissues are represented in red and slow-growing tissues in black, and those in between, the parallel fibered bone, are in purple. But I've made our species a different kind of purple because he's kind of unique. He shows this parallel fibered bone in a very large animal which would typically have faster-growing bone tissues, but he has very highly vascularized bone in terms of this laminar organization. So we don't see these laminar vascular canals in parallel fibered bone in other animals. So what we get here is a complete mix intermediate between its ancestors and its descendants. By the early Jurassic, most crocodilomorphs, crocodiliforms, were growing slowly. So people always thought that the decreased growth rates occurred after the end Triassic extinction during the early Jurassic. But we have pushed it back to at least the late Triassic. Although there are some crocodilomorphs that could still grow quickly after the extinction, we can see that the beginning of the decreased growth rates happened sometime in the Triassic. So interestingly, what I've outlined here are the crocs and the dinosaurs. These are the two major clades that survived the end Triassic mass extinction. The dinosaurs in red there are growing really, really quickly, but the crocs are growing really, really slowly. So it is probably not growth strategy that allowed both of these clades to survive the end Triassic extinction. Or if it was, because they have such different growth strategies, there is some other factor at play here. And this is what we are busy examining at the moment. So I don't have any answers for you yet because this is my current research. So if we look at today's world, where does all of this data fit in? Well, if we go back in time, we have a record of global temperature for the planet. This goes back to some 66 million years ago. And there was a Paleocene-Eocene thermal maximum, so there were spikes in temperature. And although species obviously survived, it would have been a hothouse. It wouldn't have been great. So climatologists have used this data to now predict what we might see in future. So let me zoom in on that last little bit there where we're living at the moment. And there are various projections, because as I've said, with variability, it's difficult to predict. But what climatologists are suggesting that by 2100, the climate will be similar to the Miocene climate optimum, which is something that the planet experienced about 16 million years ago, and there were no continental ice sheets then. And if conditions have not stabilized by 2250, by 2300, we may in fact see this Paleocene-Eocene hothouse world again. And this, although it's possible to live through, is really not great and is going to cause many, many extinctions because there will be multiple global warming events. And global warming doesn't just mean that places get drier and hotter, and other places, or there's a general global warming, or there's a general increase in aridity, it means that there's an increase in variability, an increase in unpredictability. So some areas that were a little bit dry may have severe drought, or they may be completely opposite and experience flooding. And we've already seen this in the last couple of years, where some places are experiencing hurricanes that are far stronger than previously recorded, or some places that were drought-stricken are suddenly having floods. And so it's very difficult to predict, and it's very difficult to adapt to a changing environment if you don't really know how it's changing. And this would also mean that there are no ice sheets at the poles. And of course, we are used to having our South and North Pole capped by ice. Now, all of that ice, if that melts into the ocean, would change the ocean chemistry. It will completely change the ocean circulation. It will cause the extinction of numerous millions of ocean organisms. And this will have a domino effect onto the terrestrial realm, where land organisms, including ourselves, would be in a lot of trouble. So what would we see? Well, from looking at past mass extinctions, we would see a decrease in biodiversity and an increase in what we call disaster taxa, such as this Lystrosaurus that we see in huge abundance after the Permian-Triassic mass extinction. What would disaster taxa look like in today's world? Mostly rodents and taxa that are used to human-dominated systems, so we're probably looking at a lot of rats and cockroaches. We're looking, yes, at a general decrease in biodiversity. And of course, biodiversity, I don't need to preach to the choir here, as biologists, is the key to a functioning ecosystem. We would also see the disappearance of large vertebrates. They tend to go extinct first, which is very sad. We already know that rhinos and elephants, for example, are threatened, but they are not likely to survive an extinction such as what we are going through. They will be the first, well, not the first to go. There have been many that have already gone, but they will not survive a mass extinction. And we will also lose our evolutionary powerhouses, our lungs, the oxygen for the planet. So the more we deplete the Amazonian forest, for example, the less oxygen is being made on the planet. And it's just silly, really. It's not very logical to be doing that, because we're almost collapsing a lung. So why do we look at past mass extinctions? Well, we need to see the consequences of the mass extinctions, and the only way to do that is to look at what's already happened in the fossil record, so that we can understand what we are seeing in the present day, so that we can try and predict some things in the future. We won't be able to predict very accurately, but we may be able to predict certain species that may survive and ones that may be more susceptible to extinction, and put in conservation efforts to try and stem the decrease in biodiversity. So my current research program is really looking at the whole of the Triassic. Now that's some 50 million years, so it's I've bitten off quite a lot to chew, but it's bounded by two mass extinctions, and there seems to be a smaller event called the Carnian pluvial extinction during the late Triassic. And I'm looking at all of the therapsids in black and all of the archosaurs in white. There was quite a big ecological reorganization during the Triassic, when the therapsids were the dominant terrestrial vertebrates, and they were replaced by the archosaurs, the dinosaurs. Although I'm looking at this more carefully, I'm looking at it for species per species instead of major group, by clade, in other words, because we do see the entrance of the first mammals at the very latest Triassic, but they're mostly only known from teeth because they're so small, they're the size of mice. So we do still see a lot of diversity in the very earliest mammals, but of course the dinosaurs sort of take over the picture. So we are now looking at this ecological reorganization through the Triassic to see how mass extinctions affect the vertebrates going through them. Thank you.
Thanks, Jennifer. Any questions from the audience or online?
Just saying time. I don't think we have time.
I can ask one.
Thank you.
You talked about the growth rate of these large sauropods, and you talked about very rapid deposition of bone. But that also linked to longevity. I mean, if you live longer in indeterminate growth, you're going to get bigger. Is that something to have considered?
Well, we're actually finding that all vertebrates have determinate growth. So you do stop growing at some point. And although the large dinosaurs would have taken sometimes 15 or more years to reach maximum size, that's actually pretty quick in terms of the size that we're talking about if we're talking about a 70-ton dinosaur. So we do know that the very large theropod dinosaurs and the large sauropods, they did grow quickly to reach at least reproductive maturity and then maximum size after that. But they do stop growing. Even crocodilians stop growing. So we have found evidence that even though it takes longer in something like a crocodilian, every vertebrate seems to have determinate growth. So none of the vertebrates that we have studied on the planet really have shown true indeterminate growth. They're just dying before they've stopped growing. Thanks.
Thanks very much, Jennifer. You've given us a picture of the future that looks like everything can get big or small or disappear, so it's one or the other. I'd like to thank the three speakers that have provided us with different insights just before the lunch break, and a round of applause for the three speakers. Thank you. So please join us for lunch outside, and we reconvene at, I think it's 15 minutes to 2, is it right? Yeah. So, and thanks again. for our symposium, and it's good to see the crowd back. And to kick off the final part, I have Professor Glynis Goodman from the University of Witwatersrand in our school, and it's a great pleasure to have Glynis here after she recently was promoted, just for everyone's information. So, and that's an important a milestone in her career and an important part of what we do in our school. Glynis will be speaking on the origin and diversification of everlasting daisies, Helichrysum, and has its arid origins pre-adapted it for an alpine existence. Glynis, thank you very much, and over to you.
Thanks very much, Neville, and thanks for the honor of participating in this tribute to Professor Ballinsky. We have 2 talks now on plants, and it was really lovely yesterday to see that Professor Ballinsky loved his garden and was very active there. So, yes, I'm talking about an evolutionary aspect of Helichrysum, and I'm going to start off just by reminding you how biodiverse South Africa is. It has about 23,400 flowering plant species. That makes it the 6th most species-rich country in the world, and certainly the most species-rich on the African continent. In addition, 9 different biomes have been recognized. So with all this diversity in our country, there must have been adaptation to new climates, soils, other vegetation types over time. And the question is how? Obviously, natural selection has played a role. But what about pre-adaptation? In other words, traits that already exist in an organism, playing a role in helping it to survive and flourish in a new environment. Or even exaptation, where a trait is taken from one environment and changed in a new environment to help it to exist in that one. The function is changed, the trait remains the same. So Helichrysum offers us the opportunity to look at that scenario. It's the 2nd largest genus in South Africa, and there are about 600 species worldwide. We have about 245 in South Africa. It's found across Africa, Madagascar, the Greater Mediterranean and Eurasian regions. And it's been shown to be polyphyletic and paraphyletic. It has a number of other genera in it. Some are listed here. And collectively, they are known as the hapclade. The most species-rich regions in South Africa for Helichrysum are the Drakensberg Mountain Centre, and then to the north of that, you'll see another area of red, and that is what is now called the Limpopo, Mpumalanga, Eswatini escarpment area, or we used to call it as the old Transvaal, Drakensberg, or the northern escarpment of the Drakensberg. It also has the highest areas of phylogenetic diversity, which is a reflection of the evolutionary changes that have happened in those regions, the amount of speciation. So within the Drakensberg Mountain Centre, there are 102 species of Helichrysum, with many of those endemic or near endemic. And this prompted me to be really interested in looking at the role of the Drakensberg in speciation within South Africa. And so we initiated a project where we created a dated phylogeny for Helichrysum, for the hapclade, because we needed to include representatives of the other genera that are still named as such. And to date it, we included a whole lot of outgroup Asteraceae. So eventually we had about 328 taxa, which are not all shown here. This is essentially the hapclade. And we used 2 fossil pollen dates for dating it, and then a mean of 3 studies for the base of the tribe to which Helichrysum belonged. We also did a a biogeographic analysis using a program called RASP, RASP, Reconstructing Ancestral States program from phylogenies, and we allocated eventually 9 geographic regions. We had more, but because of the complexity of the analyses, we had to reduce them and combine some of them. What this showed us is that the hapclade and helichrysum originated in the succulent Karoo about 26 million years ago. And very interestingly, in the first main clade there is migration to the Drakensberg mountain center. So the succulent Karoo is shown in the orangey red color, and the medium green there that's been circled includes some of the taxa from the Drakensberg mountain center. And what was really fascinating is that those are some of the alpine species. And so we started to wonder, and there's another one. That is not directly from the succulent Karoo, that second one, it was briefly via the fynbos in the yellow there. So we began to wonder, were there some traits that adapted the helichrysum, pre-adapted to live in that environment, and enabled it to in a relatively short time, evolution-wise, to enable it to live in that extreme environment? The succulent Karoo is also an extreme environment. It's arid. It has summer temperatures reaching 44 degrees Celsius. Rainfall is variable, but in many regions it's very low, 20 millimeters per annum, reaching up to 300 in some areas. But of course, there is the cold Benguela current nowadays, which can supplement the moisture with some fog. And then the Drakensberg Alpine Center is also an extreme environment. It includes the Drakensberg Alpine Center, which is everything above and equal to an altitude of 2,800 meters above sea level. And just to give you an idea of what some of those helichrysums look like, so there is one clade of endemics within the Drakensberg, and most of them occur in the alpine to subalpine zones. And many of them are small, although you find some with large capitula, and they are some of the most beautiful helichrysum species that exist. So these regions are found on opposite ends of the climate spectrum, and yet they're both extreme. And is it possible that functional traits developed within the succulent Karoo that enabled these species to survive and then adapt, or perhaps not even really adapt that much more to the Drakensberg alpine region? Of course, these functional traits may function similarly in very different environments. For example, a thick indumentum reduces water loss in both places, and it would provide insulation in a very cold place. And of course, the succulent Karoo is very cold at night as well. It's very difficult to know if a functional trait is an adaptation within its environment or if it's an exaptation. So in other words, whether it has now taken on a new function. So within this study, what we aimed to do was compare functional traits of helichrysum species that are endemic to the succulent Karoo with those endemic to the Drakensberg Alpine Center, and then assess whether the succulent Karoo endemic species have traits that could have adapted them, enabled them to survive within these alpine environments. And this study was undertaken by an honours student whom I shared supervision with, Professor Sally Archibald, and I will acknowledge them at the end as well. So essentially we identified functional traits that needed to survive in each of these environments that is in the succulent Karoo and in the Drakensberg Alpine Center, measured those functional traits on Helichrysum species endemic to the two regions, and then looked for some overlap in occurrence of the functional traits in the two biomes. We measured 15 functional traits for 19 Drakensberg endemic species. These are the alpine endemic species, and 14 succulent Karoo endemic species. And we used herbarium specimens that were found in our herbarium here at Wits and also the National Herbarium in Pretoria. For the functional traits, we had 15 in total, 6 that were quantitative, and the balance were qualitative. And we measured between 3 and 5 specimens, depending on what was available. And just to give you an idea of how very similar some of these species do look. So on the, I think it's your left and my left, is the succulent Karoo species, Helichrysum desertifolia. And on the right is Helichrysum lineatum. And you would think that they both occur in very similar habitats, but they're at opposite extremes of the climate regime. So multivariate analyses were undertaken, and some box plots and t-tests were done. And if we look at the results for the quantitative data, you'll see that, if I can point here, that the internode length was much smaller in the Drakensberg Alpine Center plants. So in other words, the spaces between the leaves are much smaller. The leaves are much closer together, and obviously that would help them to trap warmth. But surprisingly, the leaves were generally larger in the Drakensberg Alpine Center than in the succulent Karoo. And it might be because a larger leaf creates a larger boundary layer, and so that also helps to trap the warmth. In terms of qualitative traits, the annual life history was only found in the succulent Karoo, which makes absolute sense because the growing season in an alpine environment is just far too short to enable it to die back every year and then grow from seed freshly in an alpine environment. Instead, in the Drakensberg Alpine Center, what you find is plants that have a rosette formation, which again would insulate the leaves within the plant canopy. There were lots of short shoots, which is exactly that, short internodes between the leaves. And as I said, surprisingly larger. They're not very big leaves, but they're larger than those in the succulent Karoo. And they also have larger capitula. And that is generally because there are very few pollinators up at those high altitudes, and so they have to be large enough to attract them and put on a good display. And then there are a number of features which actually occur in both of them. The first 3 features listed there are found equally in both areas. So 70% of species in both the succulent Karoo and the Drakensberg Alpine Center have a woolly indementum. So that is useful in both instances. They both have some woody plants. About 50% of them have woody stems, and about 50% in both areas have rolled leaf margins. Some have small leaves. The inflorescence size, about 70% of the succulent Karoo have really small capitula, as opposed to the Drakensberg Center. And then leaves circling the capitulum is also found within the succulent Karoo, more prominently with 80% of those species having that feature, which again would help to protect the flowering part of the plant, the capitulum. Dead leaf retention, which is what you see in aloes in South Africa, with the dead leaves building up at the bottom of the stem, that is found predominantly within the Drakensberg Alpine Center plants. And again, I would imagine it is an insulating factor that it would promote that. And then just to show you how different woody species can look in the two regions, although they might both be woody, Helichrysum wittbergense on the left is quite a robust shrub, and it can survive under meters of snow for months, as Stefan Grab from geography will tell you. And on the right-hand side, a very twiggy, almost sort of delicate plant in the succulent karoo, but that is also woody. So combining both quantitative and qualitative analyses, this NMDS analysis shows the convex hulls of the two regions. And what we found is that in axis 1, it was mainly leaf size and growth form that is separated out here. And once again, the annual habit, leaf size and growth form. So on the right-hand side, we have woody cushion plants, which are found in both regions. But on the left-hand side, we find the rosette formation, and it's exclusively the Drakensberg Alpine region. In the second axis, we have life history and reproductive unit size. And you'll notice at the bottom here, I don't know if you can see my arrow. No. So at the bottom is, in the orange, is mostly the succulent karoo species. And then there are areas of overlap which reflect those having similar traits. Okay. This is a cluster analysis which shows similarity and not evolutionary relationship. And what you'll notice is that there is one branch, one grouping, one cluster of exclusively Drakensberg alpine species, but the others have a mix of species. This one here has only one Drakensberg species, and that is Helichrysum lineatum, which I showed you is very morphologically similar to Helichrysum deserticola. And there it is, very, very closely matching in morphology. And then the others have a mix of Drakensberg and succulent karoo species. So what do we find so far? Well, there's an overlap in some traits in Helichrysum species endemic to each region. And these are obviously functional traits that enable survival in either of these environments. The woody cushion growth form, the woody leaf indumentum, and leaves circling the capitulum. So what does this mean? Does it mean that there's been pre-adaptation or ex-adaptation? Well, we really need to look at the phylogeny to give us that answer. And if we go back to the phylogeny, the hapclade phylogeny, which is predominantly made up of Helichrysum species, and we look at a section of it, that very first section, what you'll notice is that those alpine species which occur in the same clade are also morphologically very similar in the cluster analysis, and they're grouped together. So it would suggest that there has been a similar evolutionary origin for those features. Of course, we can't prove it, we can only infer it. But similarly, sorry, it's difficult having two pictures here. Similarly, if we look at the other clade where Helichrysum lineatum arose, and that was directly from the succulent karoo ancestors, it also falls within that cluster in the cluster analysis. And once again, that's how very similar they look. So what does this mean? Well, I would suggest that certainly there is some evidence to infer that some of those functional traits did pre-adapt Helichrysum species to live in the alpine environment. Those traits that were adapted for aridity in the succulent Karoo. And if we look at the dating, they would have started to move into the mountains at about 8 million years ago, which is when you had the cold Benguela current evolving, developing along the west coast, and aridification starting along that in that area. Of course, there could have been convergence in the two regions, and I'm sure there is some, but I think it would be unlikely that it would have happened to that extent. And then what about in the bigger picture? What about climate change? Well, if these plants have functional traits that adapt them to a very arid environment, perhaps that means that as our temperature increases and it aridifies, even the mountains get hotter and drier, that those species in the alpine environment may well be adapted to surviving in that climate as well. And so then I'd just like to acknowledge my co-investigators, Emma, Sally, and then my colleague from Kirstenbosch, with whom I created the phylogeny for the hapclade, our funders, and many others who are involved in it. And thank you for listening.
Thanks very much, Glynis. I'll first take questions from the floor, and then we'll see what's online.
Thanks, Glynis. That was lovely. Okay, I hope I'm on the right page here. Can you, when you look at the phylogeny, also see like the fynbos coming in between the Drakensberg Alpine part and the Karoo? And I was kind of wondering, where do you think the fynbos fits in between these two regions, sort of sitting maybe at like a pinnacle or whatever between those two regions? And if you started to think about some of the traits of those helichrysums as well, because I also think of the fynbos as like a very brutal environment, and maybe sort of echoing elements of the Drakensberg and then elements of the Karoo. So just was curious.
Thanks, Vanya. That is an interesting question. So the fynbos, the helichrysum in the fynbos region are mostly found on the mountains. And the mountains have a very, they're quartzitic, and so it's very poor soil. And although the basalts of the Drakensberg are theoretically nutrient-rich, it has been, Clinton Corbett has shown through translocation experiments, in fact, that the basalt soils are not releasing their nutrients at that cold temperature. So I do think that certainly if they went via the mountains in the fynbos, that it could equally well have perhaps helped them to develop some of those traits. We didn't compare it to the fynbos species, but I know that many of them are also small-leaved and probably also would survive in an alpine environment.
Just following on from what Siobhan said, so you think about what was the route of migration, and of course the succulent Karoo is a winter rainfall, the Drakensberg is summer rainfall, and you've got the Nama Karoo in between, which is summer rainfall, so that would seem a likely route, if there's a migration route as such.
So Ralph Clark has in his papers suggested that the most likely route is via the Cape Escarpment Mountains. The Neverfeldberger, the Sneeuwberger, etc. But our phylogenetic analysis didn't really show that very well. But for me, it still seems like the most likely route. So perhaps those species didn't survive in that region as traveling on a route, because I'm sure they were adapted to higher environments. I don't think they would have gone through the Nama Karoo. I may be wrong, but I don't think so. It seems much more likely that they would have gone along those mountain tops.
You've talked about climate as being the driving force here. What about herbivores?
Oh, good question. Hadn't thought about that. So I don't know that there are that many herbivores in the succulent Karoo, and there certainly aren't that many apart from the ice rats up in the high Drakensberg. And there are not that many insects either, which is why pollinators are so scarce. So perhaps the lack of herbivores is also a feature that they have in common. That's a good point. Thank you, Marcus.
This following on from Marcus's point, Glenys, and if you know that springbok migrated from the succulent Karoo into the Drakensberg and back.
Sorry, what did?
The springbok. Now, whether they carried early chrysalis seeds along the way, I don't know. But that was a common occurrence in maybe 200, 300 years ago until we stopped the migration patterns. But my question was about the size of the leaves, because I was thinking that the succulent Karoo ones had smaller leaves for water retention, but you argued the opposite, that the larger leaves in the alpine is for warmth. Is that also an adaptation for warmer climates, the smaller leaves?
Yeah, definitely. So it would. So the smaller leaf surface would definitely reduce evapotranspiration. So when I say larger leaves in the Drakensberg, I mean they're not very large. And many of the larger leaves are actually in a basal rosette, and they remain throughout the winter. But these species put up a stem region with smaller leaves, which usually carries the large flower, the large capitulum, and then that dies back every year, and it's just the larger leaves that remain, and it would have to survive under snow or in extreme conditions.
We have a question online for you, so if the person asking the question can come online. Alex, did you have a question?
Yeah, if I may. My question is it possible to trace the climate changes and phylogeny in past? I mean, how much time did it take to develop all these adaptations, and what kind of climate was in Karoo at that time?
Alec, so obviously if our dating is correct, we're looking at, as I said, from about 8 million years from when the first migration would have started to happen from the succulent Karoo. But sorry, the second part of your question?
What kind of climate the Karoo had that time?
So it would have been moister because there was more forest spread across southern Africa. But as I mentioned, it would have been starting to aridify at that stage. And presumably these plants, unless convergence happened in both areas, as I said, that's a possibility. I guess we'd need to go and find fossil plants in the region. I'm not sure how successful that would be. We even battle to find the pollen grains in the region.
Okay, I understand. The sense of the question was, if the climate was different, what was the drives which made these plants to adapt to the modern time climate? Or what was the condition, what they were adapted at the time when these adaptations appeared?
So they would have been adapting to a drier climate during the last 6-7 million years in the succulent Karoo.
Okay, so it was slow process, step by step, yeah? Not immediate appearance of something inherited from the common ancestor.
No, I think it would have been a slow process, and I certainly don't think that they jumped from the succulent Karoo to the Drakensberg either. So, it's happened over the last 8 million years.
Okay, thanks.
Thanks, Glenys. Thanks very much for your talk.
Thank you.
mic so he can show you this. Oh, sorry, I don't. Oh, which one is your presentation?
This one.
Oh, so back and forward.
Cool.
Just check if it's on. Yeah, just check it. Just test it for me. She wants the mic, but I'll introduce her. I just want to test if it's working here. Oh, there we go. Don't go away. She needs the mic. Our next speaker is Professor Kelsey Glennon from Wits University, from the School of Apes. It's good to have Kelsey presenting today. She represents the floral, the butterfly diversity in the floral sense. And Kelsey will be presenting a talk on floral color polymorphisms in the Drakensberg. Kelsey, thanks very much.
Can I just take it out? Cool. Thanks, everybody, for being here, and thanks for the opportunity to talk about my research. I am an evolutionary biologist by training, and so as an evolutionary biologist, I think a lot about variation. And when I was preparing for this seminar and learning about Ballinsky and his research, I was really surprised and quite excited to learn that he also cared a lot about butterflies. And the reason he cared about butterflies was not just because he was an entomologist, but because they have very cool wing patterns. And so what I'm showing you here is a representation of color morphs. And color is one of the most charismatic traits across the entire tree of life, essentially. Everything uses color, from invertebrates like our butterflies to herbivores or mammals, but also plants. So plants invoke color to encourage other organisms to do their job and share their gametes because they're obviously sessile. So color is a really important trait in evolution, and particularly when you're thinking about variation, because color is super variable. And not only, without a slide in front of me, I forget where I'm going and I'll talk on a tangent here. So butterflies have been one of the classic study systems for studying color variation, but flowers are another exciting system to study variation. Not only do they have an array of shapes and sizes, but as you can see here, there's a whole array of color. And so when I started thinking about color variation, just as an aside, my first love is chromosome duplications in plants and polyploids. And then I was kind of enraptured by the color variability that I had seen while I was out in the field in the Drakensberg. And so when I started studying this family, this is the Hypoxidaceae, otherwise known as the stargrass family. So this is a cosmopolitan group. I should say near cosmopolitan because they're found everywhere but Europe. And when we first started looking at them, you can see there's a few different genera that have yellow flowers. But then there are some key species that show this really cool diversity. So in the upper left-hand corner is Hypoxida. They're found on the Seychelles. And the majority of flowers are this beautiful, like orangey cream color. But then you have this inset in the upper left here, where it's more reddish black. And so why are these colors coming up? And then we have in the circles in the center there on the top is Peridia. You'll see the name Spiloxine as well, but the genus has just been revised. And so Peridia has these beautiful colors. Not only are the petals white, like I've shown here, but they can be orange, pink, or yellow. But they also have this really beautiful iridescence in the center. So there's this purple and this teal color. And so we were curious about where that's coming from. And the other thing about Hypoxidaceae that's really great is that its center of diversity is here in southern Africa. We have the most species in the genus, or in the family, sorry. But I'm going to tell you today a story about this little pink flower in the center. And this is called Rhodohypoxis. This is the genus that my group has been working on for the past couple years. And there is some color variability there. And so when we first started thinking about hypoxidaceae, we started with a macroevolutionary picture. We wanted to understand how color was distributed across the family. Was it in a single clade, or was it across the group? Did it occur in only one continent? So what was the idea here? And so when we started this, we started with using a phylogeny from Liu et al. In 2012. This is, until Monday, last Monday, the most complete phylogeny of the family. But my PhD student has just completed a phylogeny of the Southern African hypoxidaceae, but I didn't want to span it over 2 slides to show you the whole tree. So I've gone with the more abbreviated version. And so we wanted to map where color variability was in this group. And hopefully this could tell us a little bit about the transitions that underlie these colors, because the colors that we're thinking about are governed by the anthocyanin biosynthesis pathway. So there's 8 genes that underlie this. And we wanted to know how those genes are interacting, if there's differences among groups, and what we could tell from the evolution. Like, was there a shift in color, and did that spark diversification? And so what we did was we went to the herbarium and we went to the field. We collected fresh tissue, we collected herbarium tissue, and we used this super technical, really complicated technique called epidermal petal peels. And essentially what you do is you make that petal nice and hydrated, and then very carefully, this is super important, you're very careful, you use your forceps and you very carefully peel that epidermis off. Then you look at it under a microscope, okay? And so what we were able to see is the different pigments in our flowers. And so I should also tell you that we were looking for both carotenoids and anthocyanins. So carotenoids are the yellows and the oranges and sometimes reds. And then the anthocyanins are your pinks and your bright reds that you'll see. Okay, so we're thinking about those. And when you look at those pigments in an epidermal tissue, they look different under the cells or in the cells. So your carotenoids are not as compacted, and they're not as collective in the cell, and they give you a more spread out yellowish color. And in the top left picture here, you can see evidence for carotenoids. And so this is from a hypoxis flower, and we've seen those as yellow. We also found evidence for chlorophyll. So we found evidence for carotenoids and chlorophyll. So some of the white flowers, they actually end up with more of a green tinge. And this is a consequence of chlorophyll in the petal epidermal tissue. I'm going to skip the flower petal on the right-hand side for now because it's quite interesting. So I'll come back to that. And then on the bottom, we have a lot of evidence for anthocyanins. So while our petal peels indicated that we have anthocyanins and carotenoids, we really wanted to know what kind of anthocyanins we were working with, because the group that I'm going to tell you about is primarily pink. And so I'm going to come back to this top right. And so you notice a difference in the epidermal tissue, hopefully. You'll see that it looks a little bit more bubbly than the other cells that look a little bit more flat. And that's a consequence of structural color. And so I told you about anthocyanins and carotenoids. And so I partnered with a colleague in the School of Chemistry so that we could use some GC mass spec to get an idea of what our pigments were between the flowers. Because there is a bit of an overlap with carotenoids and anthocyanins. So it could be that we think the flower is pink as a consequence of anthocyanins, but in reality, it's actually just a type of carotenoid. And so using GC mass spec, we found that we actually have malvidin in our pink. So there is a transition from carotenoid-based pigments to our anthocyanin-based pigments. And so this was good to know for our field work. So just a slight detour into that previous petal peel of structural color. So not only does hypoxidaceae have pigmentation, but it also uses the structure of the epidermal tissue to generate this color. And this is research that's been done by Alan Ellis at Stellenbosch University. And this is what started me thinking about color variation to begin with. And so on either sides of the screen is Paridia capensis, and you'll notice on the left-hand side it's yellow, and on the right-hand side it's white, but they both have that beautiful iridescence in the center. And so this color is non-pigment based, but instead it's a consequence of the epidermal cell shape, and that's what lends itself to the iridescence. Okay, so back to our phylogeny. So now we've got an idea of what the colors of the different species were, and then we plotted them across the phylogeny. We've done not only color, but also continent, because we wanted to know, is all the color here in South Africa, or is color spread across the globe? And what we found was that we do harbor the majority of the color variation here in South Africa. And we have multiple origins of anthocyan and coloration in the family. So this is kind of cool to figure out, because it's not just a single origin or a single transition from one biosynthesis pathway to another, but this transition has happened multiple times in the family. Here I have 8 different transitions that have been noted. And that's a lot of work for a group to go through. So there's some selection pressures here that are acting through time that are sparking these transitions. I was a little bit disappointed to find that color variation didn't necessarily lead to diversification. I was hoping that one clade would have all of the color in it and be like, oh, cool, color is responsible for the species diversity. But I'm still happy with the multiple origins that we found. Okay, so I've given you an overview of the family and how color the colors transitioned among the different genera. But what I'd really like to tell you about is this little genus, and particularly this species that I love so dearly, is Rhodohypoxis bowerii var. Confecta. So the species Rhodohypoxis bowerii has 3 different varieties, and I've pictured them here. So in the upper left is Platypetala, and this variety is predominantly white. You might be lucky and find a few pink morphs in the field if you spend enough time at the different populations. And I want you to remember that it grows in a shallow, rocky outcrop environment, okay? Then we have bowerii, the variety bowerii. These are bright pink. They're beautiful little flowers that grow throughout the Drakensberg. So you've already been introduced to the Drakensberg by Glynis, so hopefully you remember the mountains. We're going to spend some time there. Bowerii grows in much more marshy and damper environments than Platypetala. Okay, so you remember the 2 distinguishing features there ecologically. They also span the length of the Drakensberg, so from the northern parts of Golden Gate all the way down into the Eastern Cape. Okay, and bowerii is focused primarily in the Eastern Cape, where Platypetala is across the entire range. So if you go to Golden Gate or you go down to the Drakensberg, you'll find platy. You might have to look a little bit harder for bowerii. If you're really looking at the tops of the peaks, you'll find Confecta. So Confecta is the 3rd variety. It was described by Hilliard and Burt in 1979, and they named it Confecta because it looked like confetti was sprinkled across the mountaintops. And so you can see how it has these beautiful white, light pink, and dark pink flowers. Okay? And so Confecta is only found at the high altitude populations. Okay, so you remember the 3 differences ecologically between the 3 groups. So Platypetala, a little bit drier, rocky soil, predominantly white. Bowerii is marshy and pink, and Confecta is at high altitudes. All right? And so when we were thinking about this group, we wanted to know whether or not there is any potential for diversification or selection driving these 3 varieties into being novel evolutionary lineages. And color seemed to be an obvious trait to look at for this question. And why is color an obvious trait? Because there's really nice selection agents. So when we think about evolution, we think about variation, and then we think about how selection acts on that variation. And flower color has these 2 primary groups of selection agents. First, we have the biotic, or your pollinators, and then second, we have your abiotic, or your environmental variables. And so these are 2 really nice classic studies about how you can compare biotic and abiotic selection agents. Linanthus perrier is a famous example of Sewall Wright being wrong, because this was an example that he published in 1930 where he said the color morph variation was due to drift. And so that would have been the first example of drift or random changes in a population in most of plant sciences. But then Shemsky and Bierdajak went back a couple decades later and surveyed the populations over a decade. And they measured environmental variables, pollinators, etc. And what they actually found was that the color morphs, the differences in the numbers between purple and white morphs, was due to spring precipitation. So Wright was unfortunately wrong, and this is not drift, but instead an example of natural selection. And I like it because it's an example of abiotic selection agents, and there's not many of those if you go through the flower color literature, because everybody loves pollinators. On the right-hand side, hopefully you recognize the snapdragon. This is a phenomenally exciting model organism for flower color evolution in plants, because you've seen them in the shops. They're marbled, they're bright pinks, they have little, you can make the dragon lips orange if you want. We know a lot about not only the colors that you can get, but the genetics that underlie them. And so the snapdragon, their color, before we started controlling it genetically, is a consequence of pollinators. So you have two examples of biotic and abiotic selection agents. And so we wanted to know with Confecta, where do we lie? Do we have abiotic or do we have biotic selection agents? So our first question was, are pollinators our selection agents? It took us a long time to answer this question. Every time I'd give a talk about flower color, I would willfully ignore the fact that pollinators existed, and then somebody would ask, well, what about pollinators? And now I can finally answer this question after many hours in the field catching insects, often through the night, because maybe they were also out at night. And we found that these 8 insects total, just these, and they're now dead, interacted with our flowers. And so there's really not strong evidence for pollinator interactions with them. One of my former master's students, Bianca Ferreira, painstakingly brushed all of the pollen off of these insects and compared it to our Rhodohypoxis pollen to find that indeed only 4 of these insects actually had any solid pollen load, and we think that they had been blown onto the flowers and were escaping the wind. So they didn't really intend to do any pollinator acts at all. So the other thing that we needed to think about, because pollinators are key for reproduction in plants, but plants are very wily creatures in that they don't necessarily rely on other organisms all the time. And so we wanted to know if Rhodohypoxis was just as wily and whether or not it would rely on pollinators exclusively, which seemed like a bad choice given the 8 insects that I've just shown you over multiple hours. And we wanted to test if they were asexually reproducing. We'd go out into the field, we'd collect these, what we thought were single plants, put them in a pot, and the next season we'd have like 1 million plants in the same pot. So either something in our greenhouse is an extremely effective pollinator and the seeds are being dispersed right into the pots, or there's clonal reproduction. And so we dug up our plants and we found that there are in fact little pups growing off of our plants. So Confecta is taking part to some extent, not exclusively, but some extent, to clonal reproduction. The other thing we wanted to look at is whether or not they could self, because plants are incredible in their reproductive strategies. So they don't invoke just one. In some instances, they may take on 2. And so this is a cross-section of a Confecta flower. You can see the stigma on the bottom. It's this little wavy thing, and it's about 1 millimeter tall. It's very small. Sorry for the no scale bar. But it looks like a miniature baobab tree stuck in the base of our flower. And you can see the anthers. Those are dehisced anthers, where they're open, and they're just crowded over those stigmas, dumping pollen on top of them. So self-pollen can happen in plants, but it doesn't necessarily translate to seeds. So we did hand cross-pollination experiments to test whether or not they are capable of selfing, and it turns out Confecta is not only happy to minimally produce little pups via vegetative reproduction, but it's also quite happy to self. It's one of my most prolific selfers in the greenhouse. So this just highlighted that there's unlikely to be strong selection pressures from pollinators on flower color, because if there's no movement of if the plants are able to reproduce without pollinators, why entice them to see you in the first place? So that left us with our second question of whether or not abiotic variables or the environment affected flower color variation. And just to give you an idea of how expansive for this tiny flower, this is a relatively expansive range across the Drakensberg. The white circles are Platypetala, the pink circles are Bowerii, and then those half circles are Confecta. And we focused on this one population in the Free State. So we went to Sentinel Peak, and you can see the picture of Sentinel Peak on the right-hand side here. This is taken from the Vitzi's Hook Lodge, where we set up our little research station. And we worked at the base of Sentinel Peak at the car park to kind of get a better idea of what the environment was like for these variable populations. And so what we set out to learn, one thing I forgot to tell you about Hilliard and Burt's hypothesis with Confecta is that when Hilliard and Burt described the species, they hypothesized that an individual flower started out white, then went to pink, and then went to bright pink throughout the flowering season. And that seemed like, we were seeing evidence of some transition in the population through time, but we weren't necessarily sure if we agreed that it was the flower itself or maybe the plants. So we tested whether or not the flowers or the plants transitioned through colors, and then we asked about UV radiation and soil moisture. And no study on selection is complete without measuring fitness. So we did estimate seed count and germination for the different morphs to figure out who might win in this evolutionary battle. So when we worked at Sentinel Peak, we worked with Prof. Sandy Steenhuisen and her postdoc Nick Lemaitre, and we first went out and tagged multiple plants, between 40 and 50 individuals. We lost a few, and we went and photographed them. Well, Nick photographed them because he was right there every other day for the duration of the flower's life. So the flowers themselves live for about 2 weeks, and then the plants themselves are out from about October after the first rains all the way through until about January. So Nick took a lot of photographs for us. So he photographed not only flowers, but also the plants through time. And so here is a picture of Promise, who ultimately took this project on for his master's, with Sandy photographing our flowers with a color swatch so that we could kind of calibrate how we measure flower color change through time. And so the next 3 panels are pictures of the same individual at 3 different time points, and you'll notice that the individual flowers don't change. So we didn't see any evidence that the flowers themselves would change through time. However, we did find some individuals that would put up a nice, beautiful white flower in the beginning of the season, and then towards the end of the season would put up a nice, bright pink flower. And so then we started to wonder, okay, my title is a bit of a misnomer because this isn't exactly the definition of a flower color polymorphism, as we would call it in the literature. So the flowers are not polymorphic per se, but they're instead probably an example of plasticity. So it might be that plasticity is responsible for this variation that we see. And so following on with our fitness, we wanted to know whether or not white flowers or pink flowers were more fit in the population. So we tagged individuals that we knew, okay, this was a white flower. We waited for it to set seed, then we collected seeds if it did. If it didn't, then we recorded it. Our white flowers were really prolific in their seed production, but our pink flowers had much better germination. So 30% of our pink flower seeds germinated versus 8% of our white flowered seeds. And there's 2 reasons that this might be the case. One is that the pink flowers are coming out later in the season, so they have much less time to get themselves established before winter sets in. Sometimes these seeds germinate immediately, and some of them will wait for the season. So if you're an immediate germinator, you need to start working hard before the winter comes and you freeze. The other possibility is that Pigmented or pink flowers tend to have a higher internal temperature, so they're warmer for the seeds or for the pollen, and then also that might speed up development, which then leads to faster seed germination. And so there might be this thermal link with anthocyanin production that's facilitated this faster germination. But right now, it seems that our pink flowers are better at germinating than our white flowers, though given their propensity for selfing and clonal reproduction, it may not ultimately matter. And so when we're thinking about our environmental variables, we had 3 sites at Sentinel Peak that we looked at. So first we started at the car park, then we had a site at the lodge, and then we found a catchment off the side of the mountain where we counted morphs in square meter grids. We took estimates of soil moisture and temperature as well. So right in the beginning, we wanted to know what the populations themselves were looking like with environmental data. We subsequently downloaded UV data from NASA to test whether or not there was a difference there. But the next graph I'm going to show you is just based on what environmental data we collected in the field. And so we found that as soil moisture increased, so did the number of pink morphs. Within rhodohypoxis, this makes sense. Within the greater scope of the flower color literature, this did not make sense. And we expected to find the opposite pattern. So we expected that the wetter conditions would be more stressful and that you would find more white morphs without the anthocyanins. But that wasn't actually the case. And so the catchment and the lodge sites are the 2 bottom lines. And you can see there is not really any change in pigmentation or number of flowers there. I have another hypothesis as to why that's the case, but I'll leave that for another day. But if you look at the top line, that is our car park. And so as you move through the season, you'll notice that there's A, more rain because we're going through the rainy season, but B, that there's more pink morphs. There's also an altitudinal difference between each of these 3 sites. And so there's likely a complicating factor with altitude, and that might be the UV, because the UV is increasing as you're moving up in altitude. And so it's possible that UV might be kind of driving this pattern and we're only picking it up a little bit with the soil moisture. And so for Promise's MSc, we did some growth chamber experiments where he could control UV exposure and soil moisture. So he used 4 different treatments. Our plants are very small, so they looked very silly in our very large, amazing environs. And so throughout 8 weeks, he measured traits, both number of leaves, the length of leaves, the number of flowers, and of course the color of flowers that emerged. And so what Promise found was that the plants that were exposed to UV had more and longer leaves, and they had more flowers and slightly more pink flowers. So if you look at the bottom right graph, you'll see that the percent of pigmented flowers in UV treatments was trending towards being greater, but it was not statistically significant. And I think that might have been because our sample size was a little low. So we're going with the idea that UV is generating more pink flowers. This does make sense biologically. And so what we learned here was that Confecta's color variation is likely plastic. So the plant itself can produce a white flower morph in the early part of the season. And then as the season gets more stressful, there's more rain, there's more sunlight, they start to put out these pink morphs. And so remember I told you that the flowers are pigmented as a consequence of anthocyanins. So anthocyanins are really useful for plants in stress responses. And so as the soil moisture gets higher, these are little geophytes, and so they have little bulbs in the soil. A lot of wet ground around a bulb is not necessarily the best thing for it. And so the production of anthocyanins might be a compensation for an increasingly stressful environment. And then the solar radiation, as the sun is moving across the berg through the summer, where you see the increase in the pink flowers tends to get the brightest sun and the most sun. And so these plants might actually be using the anthocyanins as sunscreen to protect themselves through time, because it doesn't really seem like there's an evolutionarily diversification reason for having these morphs in the population, but instead it's probably a consequence of the environment and them protecting themselves. And so with that, I'd like to thank the members of the Peas Lab, both past and present, because without them, I wouldn't be able to present this data to you. My colleagues at Kwakwa, Sandy, and Nick, and then Sadhna at Chemistry, and then the funding that made this possible. And thank you.
Plants with sunscreen, that's what I got. Are there any questions online or from the audience? We'll take one or two because we almost out of time.
Kelsey, are the seeds, or do they have dormancy?
Yes and no. So we've noticed that some of our seeds will sit in an envelope very happily through a couple years and then germinate, and then some seeds will germinate right away. So yes and no.
That's pretty normal.
Thanks. We have a question from Mary Scholes online. Mary?
So, hi, Kelsey. Thank you very much. Nice to see you and the experiments. So, Kelsey, just in case you don't know, they've put in a fully automated weather station at Sentinel so that you're able to now get up-to-date weather data. But what I really wanted to ask was, have you measured the level of anthocyanins? Because I've never seen anthocyanins as a pink. I usually see them as a deep maroon color that are uncondensed tannins.
Thanks, Mary. So we have not yet measured the quantity of anthocyanins in the petals, but it is on the cards for the future. So hopefully I'll have an answer for you soon.
Thank you. Great talk.
Thank you.
Thanks, Kelsey. That's all the time we have, but thank you very much. Our next two presenters, I'm not sure which one is going to present, is Dr. Roman Svitin and Dr. Yaroslav Sirota. And they're both from the Smerlazhin Institute of Zoology in Ukraine. And there's an association with the Northwest University in Potchefstroom through Dr. Louis Dupré. I think some of us know him. And they're going to talk about collaborative studies of parasites from southern African vertebrates. Welcome, and please introduce yourself, and we look forward to your presentation.
Good afternoon, everyone. Thank you very much for the opportunity. I hope I'm visible. Could you please tell me if you can hear me properly? Is everything working?
Yes, we can hear you.
Okay, thank you very much. I'm Roman Svitin, so I'm the one who's going to present. Louis Dupré, the famous person of our trio, is currently on the plane, so he will watch the video later. May I ask if the presentation is sharing correctly? Is it full screen mode or?
It's full screen now.
Okay, thank you very much. Just before I start, while you are seeing the picture of a hippo standing on a piece of Lego that his kids drop on the bottom, I just have to say a few little things. So I I hope you all had a very nice and tasty lunch, and there are gonna be some photos associated with parasites and ways of they're collecting in the presentation. So if anyone have troubles with keeping lunch where it belongs and watching parasite pictures at the same time, I really have to warn you in advance. And the second thing that, of course, collaborative studies of parasites between Ukrainian and South African scientists didn't start it with only people that I'm gonna mention today, but I just wanted to share a few stories of events I was a part of. And yeah, I've seen a lot of very scientific topics today, so I have to dilute them with my just full of photographs presentation. So let me introduce you the person whose fault was it and how all that story started. This is Dr. Alena Kudlai. She is a former, or maybe she's still appointed as a researcher at our institute in Kiev, the Schmalhausen Institute of Zoology. And not that many, but years ago, she was at the conference in Barcelona and she met Professor Nico Smith. Maybe some of you know him. He's a professor, he's the leader of the water research group in Northwest University in Potchefstroom. And he was so impressed by Alena's talk that he invited her to do a postdoc in Northwest University. So she agreed, and there were a lot of promises that it all gonna be nice and adventurous and pleasurable experience. And when she just arrived there, we've been talking, and she told me that it was really nice. So I'm gonna share a few pictures from the field trips. I know that most of presenters shared some nice pictures from nice landscapes of Africa. So I don't know how good is the screen there, how much you can see, but Professor Nico is sitting on the chair and Alena is here, and I don't know how good is the screen, but that the trailer parked with the inside the trailer there's a beer, so it all started like really a big piece of fun. So they were chilling, they were collecting fish, which was the primary object for parasite studying. They were collecting some vertebrates, and then obviously they had to dissect them to remove some parasites. So they were also enjoying that dissection part, at least at the beginning, and it was more and more enjoyable over time, and then some more dissection, and then some more dissection, and it was not really that easy with the time, and there was some more dissection. They've been dissecting all the different species of different fishes and dissecting, and there are still so many photographs to go, but I don't think that I can overcome my limit on juicy photographs of corpses. But what were they looking for? So Dr. Alena Kudlai's primary focus was trematodes. The trematodes, they are very common in African fishes and invertebrates as well. So you can see how nice and juicy are they. So most of these photographs are taken directly in the field, and there are multiple places where the parasite is penetrating the fish. And this is how it looks inside, or it can be a little bit bigger. So there are larval stages, different stages of development, including the adult trematodes. And I know it's not for everyone, but many of us consider trematodes beautiful. They truly are. They can live in all different parts of fish organs, and Alena and colleagues, they recovered them from muscles, from organs, from gills, from eyeballs, from brain, and they truly are beautiful if you look closer on them. So these are photographs, microphotographs from different microscopes. These are larvae that they recovered from eyes and brains of the fish. So these are larvae that trematodes that parasitize birds when they are adult, and these are fish-eating birds. And the parasite really care about the bird, so it wants bird to be well fed. That's why it often goes to the eyeball or goes to the brain and make fish way less careful when the bird is approaching. So the adults probably parasitize cormorants or some fish eagles or some other birds that they could never get permission to dissect, but at least they could work with fishes and they could study these beautiful creatures, and they could draw different conclusions on the bird migration, on the fish migrations. They did marine fishes, they did freshwater fishes, they built nice phylogenetic trees that I also saw people were sharing, and there were a lot of questions about phylogenetic trees. So these trees are also full of questions, but they also give some answers. So it was really interesting. And it was so interesting and exciting that there were students who were willing to join Elena in her research. These are very talented and hardworking students. And as you could see on previous slides, that hardworking is the requirement for collecting these parasites, because fish is not the thing that you can halfly dissect and leave for tomorrow and continue, because by tomorrow in South African climate, you really have to block your nose with something to keep dissecting. So that's why you really have to finish it all in time. So Anya Vermak and Corette, I'm sorry, I'm very terrible with pronunciation of difficult South African surnames, so I'll just keep it typed. They started work with Elena. They published several papers. They both still in academia, and they doing their PhDs. And Elena is not in South Africa for a few years already, but they keep visiting her in Europe, and they still working together, and it all just works as a well-oiled machine. Yeah, obviously students are also dissecting, and now they do it even without Elena, and they working, but I promised I'm not overcome my limit for dead animals photographs.
That was much anticipated.
I'm sorry, is there something that I should hear, or should I continue?
Please continue.
Oh, okay, thank you. Okay, so the next point was studying of other group of parasites, which I'm personally working with. And how did I end up in South Africa? The answer is very simple. So Elena and I are friends. And luckily, Elena worked on the third floor, and on the second floor, in the same Northwest University, still working Louis Dupré, the very famous professor in South Africa. Many know him as a frog guy, and he's leader of African Amphibian Conservation and Research Group. And he's been all over the news very recently for the description of the new bullfrog species. And I talked to him over the phone yesterday, so he apologized for not being able to participate in person this conference. And he told me that he, like just yesterday, he's been citing Ballinsky in one of his paper, because apparently Ballinsky's been working with bullfrogs as well. He's really, wide-minded person. So Prof. Louis was looking for a postdoc to work with nematodes from amphibians, and he couldn't find one, and he just started asking people around. And one happy day he bumped on Elena and asked her if she knows someone who's working with nematodes and would like to come to South Africa. And just by happy coincidence, I just finished my PhD and I came to South Africa to work with nematodes, where I spent one of the best 4.5 years of my life. So what did I do there? Actually, it's not much of a difference. between collecting trematodes and nematodes. I just expanded my host list to reptiles and amphibians, and we've been dissecting, and we've been collecting nematodes. We've been collecting nematodes from literally every species we've been working with. They are so common in South Africa, and in my personal opinion, they are the most beautiful animals in the world. I don't know who can disagree, but maybe there are some people like this. They are a little tricky to study. That's why people are avoiding them, because they are too boring with their morphology, but it's like basically a hose pipe in a hose pipe, and even living in a hose pipe if we're talking about and testing nematodes. But they can also tell us different stories, and I'm gonna tell this one particular. And this story starts with another very passionate student at the time. Now it's Dr. Edward Netherlands. At that time, he was PhD student Ed, and he really wanted to study a life cycle of blood parasites. But the blood parasites he's been working with, they are like super difficult, and many intermediate hosts were not known. But he found some nematodes that have a blight parasitizing stage, and his energy has been shared everywhere around, and it touched me as well. So together with Ed, we tried to study a life cycle of a very interesting nematode. So this is the life cycle. The nematode is parasitizing in a toad on an adult stage, so they are really long as normal nematodes. They live in the body cavity of guttural toad, one of the very common species. On a very short video, you could probably see how it looks like. We've collected toads, this toad specifically from Sodwana, so we thought we will enjoy our field trip on a shore in Sodwana, but we didn't even have time to look at the ocean. We saw it from the car window, just a couple minutes, and then we had to go back to work. So the adult ones are parasitizing in a frog, just in the body cavity. But to be able to transfer from one frog to another, they have to use mosquito. In order to use mosquito, the female nematodes produce microfilaria in the bloodstream. This is how they look like under the microscope when they stained, so they're not all that purple. So mosquitoes are really like to bite this toad, but it was not also that easy. The first thing that Ed noticed, that only males of frogs were infected with microfilaria in the blood. We've dissected just a couple males to collect adults. We didn't kill most of the frogs. We just took a little bit of blood. And we were curious why only males. And the males were calling, they were using their territorial calls, not mating because it was not the season. And mosquitoes were, as you can see on this one, very happy to see them. So we collected mosquitoes directly from the toad, and we've studied all the different stages, development of mosquito. One of them is called the sausage stage, or sausage-shaped larvae. That's important thing to notice. The sausage is playing this role in the species as well. So eventually it grows in the mosquito up to infective larvae and wait for mosquito to bite in the next frog. And we couldn't make mosquito to bite frog. I mean, for people, for other animals, it's so easy. You just put mosquito next to the skin and it's biting and sucking blood and everything is working. But these mosquitoes, they didn't want it. So I told Ed, let's keep them thirsty. So let's not give mosquito any drop of water for a day, and then we'll see they will have no choice. They will have to drink something, they will drink blood. Instead of getting thirsty, all the mosquitoes just protested and died. We spent 2 nights like the whole night collecting these bloody mosquitoes, and they all been dead like within 10 hours or so. We collected them again, and then we realized that mosquitoes are attacking, they bite in frog only when the frog is calling. So when the frog is croaking, making the sound, that's the only moment when mosquitoes are coming and start take a blood meal. And when we did our experiments back in the lab, we realized why is it? Because the moment the frog stopped calling, it just turned back and started collecting these mosquitoes that are around. So it started eating them. So mosquitoes are too smart, and they were biting frog only when it's been busy with hormones and had thoughts about other things, not eating them. I said that sausage is going to play an important role. It appeared that the adult nematodes, they belong to the new species and even to the new genus. So we described a new genus, and we named the species after the famous South African sausage. So this nematode is known as Neophylaelides boerewors. And the name boerewors was also liked by many South Africans, so Ed also became famous. He's been giving talks on the radio, different shows, and we really enjoyed this part of experiment, especially afterwards, but it cost us many sleepless nights. I could speak about nematodes like for a really long time, and as this frog, I can see them everywhere. But I also would like to give the credit of for four wonderful students I had honor to work with. I wasn't supervisor for all of them, just for one I was the supervisor, for two I was co-supervisor, and for one we just shared some research, but we have papers together, and I'm very happy. I'm still in touch with all of these wonderful people. They all are successful, some in academia, some in business, and I'm very glad that studying of nematodes really help you to work on your career. And another thing about nematodes, there's so many of them, and they are so interesting that I couldn't do it just me and students, so I really needed advice from big professors. So I did what I could, and with help of Prof. Louis, we've invited my PhD supervisor, Dr. Yuri Kuzmin from the same Schmalhausen Institute of Zoology. And he could come for only 3 weeks. So people who know him know he's very passionate photographer. So everyone would think that the thing he's been doing in South Africa is taking photographs of nice animals, but the truth is, as all of us, he's been dissecting and collecting. And we picked him up at Johannesburg airport on our way to the field trip. So he didn't even have the time to drop his suitcase in the room. He's been involved in the work just at the beginning, and yes, his help was really, really big, and he still is, with describing numerous species of nematodes, and there's just some photographs of 2 of them, and I think with his authorship we already described 7 new species of nematodes from South Africa. There are still several to go. And when my time in South Africa was close to the end, Prof. Louis said that, and Olena was already gone, so Prof. Louis said that concentration of Ukrainians in Northwest University is dropping, and then we really need someone else. And he asked me if I know a person, so I said, of course I do. And there's another parasitologist from our Schmalhausen Institute of Zoology, Dr. Yaroslav Sirota. Come on, there should be a name. So he's been brought to South Africa to have some fun, which was mostly collecting and dissecting and keep working. So nothing new here, but the new is he started to work with cestodes. These are flatworms. The funny thing that we've scarcely noticed them when we collected. Olena almost never came across them, and I almost never found them. But the moment Yaroslav arrived, we started collecting them in very, very common. Okay, sorry. The cestodes are even more difficult than nematodes to work with because in South Africa there were so scarce studies and there no head start. And in my opinion is expressed via text on this slide. And Yaroslav was also not a very big fan of just boring describing new species. He's also the guru of numbers and statistics and programming. So he said that we should do a database for all the parasites we are working with. And we said, yes, definitely, we should have a database. And he said, okay, I'm gonna make it. So he created a database and it's called Parasite. And the database is still running. Yaroslav is also not in South Africa anymore, but the database is very useful, friendly, very nice, and it's working, and everyone are very happy with it. And I saw that all the previous topics were so scientific, there was so much of interesting research, and I thought that with all these photographs of dissecting and collecting might make an impression that we were not doing science, we were just having fun. But the truth is we've actually, oh, come on, sorry. We've actually did a lot of research. We've published quite a number of papers. Altogether, it's close to 100 papers in good journals, and we participated in numerous conferences in Australia, in United States, in Europe, in South Korea, and of course, numerous nice conferences in South Africa. Our data was presented in many conferences in Cape, in Kruger Park, one of the most favorite conferences, and there were people from all over the world. So we really, really extended our collaboration with other universities and institutions and other countries, and it all still running. And just if not everyone convinced that studying parasites is very scientific and perspective, just to notice that the people who worked with nematodes from Africa already got a Nobel Prize almost 10 years ago, so it's probably soon the next Nobel Prize for parasites should be coming. So if there are people who are interested to work with parasites, I'm sure there is an open spot. I would really like to say a huge thank you to so many people who were helping during my time in South Africa. I would also like to thank the Ukrainian team, the Institute of Zoology, the director, Vitaly Harchenko, who is here, who was very kind to let go me and Yaroslav to South Africa. And I, of course, want to thank Armed Forces of Ukraine for being able to present and even still having the electricity today, even after that shelling that happened today's morning. And there are numerous organizations involved, our institutions in Ukraine and in South Africa. And my big thanks to them as well. And thank you everyone for attention. That's the only photograph where all three of authors of this presentation are present, but we're also here with people there are people from South Africa, from Ukraine, from United States, from France, and we're all taking photographs in Mozambique. So our collaboration is really beyond only Ukraine and South Africa. So thank you very much for the opportunity. And that's it from me. Thank you.
Thanks you very much, Roman. I'm going to ask for questions off the floor and then online. thought there would at least be a Boris question somewhere. But, but I think there's a question online.
Mm-hmm. Yes, it's a question about if we use the DNA barcoding for nematology. It is using the tricky thing with nematodes, with parasitic especially. They are so diverse. So for two different groups of nematodes, the difference in the same gene, for one group, like 1% difference in CO1 is going to be interspecific variation. For some group, it can be a difference between different genera. And for each family of nematodes, you really have to find a properly worked barcoding gene. And there's so few data in GenBank. So we are trying to do this. We're implementing CO1 for some of our nematodes. It is definitely possible, but it's like at the very, very beginning stage. There were attempts to use 18S gene for nematode barcoding in Madagascar, but it's just too conservative for some nematodes, and it just didn't work. Sorry, my video dropped. So the short answer is yes. It is possible. We do have experience, but it just not running it.
I'm Marcus Byrne. I'd like to say thanks for a very entertaining lecture.
Thank you.
I'll look at my food differently tonight. What I wanted to ask was, do you record any pathology associated with any of these infestations? Because it strikes me that the association between the organisms is so ancient that a lot of these hosts carry the parasite with almost no pathology.
So for my personal group, for nematodes, the great majority of them are like close to harmless. At least hosts, the frogs and toads and turtles, they didn't show any complaints. Even there are nematodes that parasitize in lungs and they feed in on blood directly from a lung. They bite the piece of lung and suck in blood. And there can be 100 of nematodes in a frog's lung, and the frog keep jumping and it doesn't breathe heavily. For trematodes, the ones at the beginning, they mostly use frogs and fish and our target hosts as intermediate ones. And because they want the definitive host to eat the intermediate, then they do a lot of harm. They can damage organs, they damage badly nervous system to make sure that fish is... And if you notice the catfishes that swim and gulp some air on the top, even though the water is clear and there's definitely enough of oxygen in this water, very often is because their brain is full of diplostomatid trematodes, and they wouldn't need to come and take the air from the top, but the trematode telling them you just really have to swim on the top of the water to be available for fish, for birds. Sorry. So there are some pathology for some species, but mostly, no, they are exactly as you said, long evolution relationships made them like each other but more.
Thanks very much, Roman. I've really encountered someone who loves parasites as much as you do, but it was very entertaining. And thank you again.
Thank you very much.
And all the
Thank
best.
you. Thank you. Bye-bye.
Okay, it's the last speaker for the day, and it's my pleasure to introduce Alexei Bolderiev, And I know Alexei just from interacting with him online, and it will be nice to hear him speak now on his topic, the development of neuroscience and electrophysiology in Ukraine, from Ballinsky's contemporaries to modern neurogeneration research. This is a bit different to parasites. So Alexei, over to you, and let's hear what you have to say.
Thank you very much. So I will try to do it as simple as possible, and it will be quite historical thing. Do you see? Yes, you see my my screen. So I'm a neuroscientist myself. I'm a neurophysiologist, and I will tell like several stories about what we are doing in Ukraine. And of course, I should start with Boris Ballinsky and how did I come to know him. So it was 1991, as we knew recently that Boris Ballinsky was in Ukraine. So this picture is from 1993. I don't know, no pictures from 1991. But still in 1991 I couldn't know about him because I was a small boy. And but today's morning I remember two things about myself from 1991. It was just the summer before the collapse of Soviet Union and Ukraine becoming independent. I was reading a book at that time, and this book was Le Capitaine Cassicou from Louis Boussinard, and this is about South Africa, and it's about Anglo-Boer War. And I was 8 years old, and I learned these old names. Most of them changed. I know, like Kimberley, Bloemfontein, Ladysmith. I remember them for my whole life. So, firstly, I got acquainted with South Africa, and I'm really pleased and honored to speak to your audience. And the other thing I remembered from that summer, that I caught this butterfly. So it's one of the Papilionidae. So it's called Podalirius. So it's common for Europe and Asia, and I believe in Northern Africa also. But of course, yes, this is like, you know, like some fatum. I'm a little bit fatalist that it happened to me. And then when I was already a researcher, young researcher in neurophysiology, it happened that I arrived in Singapore in the laboratory of Vladimir Korsh, who gave a brilliant lecture yesterday, and I'm very grateful to him for everything, for recognition of my university student interest in developmental biology. And also he brought me the story of Ballinsky I never heard before. We were just sitting under the big rambutan in his house in Singapore, and his wife told him, "Stop telling the young man these old stories." But I was very interested, and after he told me, I was digging everything, these Illinois memoirs, everything I wanted to know about Ballinsky, because it was just fascinating. And you know, coming back home in 2014, I even wrote the article on Wikipedia about Wits University, so you can check it. It is in Ukrainian. Also, it's very small, because I knew nothing about you that time, but still it exists, and people in Ukraine can know something about Nobel Prize winners or famous faculty from this article. And then the war came. So the war came the same 2014, but for my city, for Kiev, it came in 2022 when missiles were striking buildings and everything was awful. And this time, my child was born just 3 weeks before, and I just opened the Ballinsky memoirs to see what did he feel at that time. So of course he wrote it many years after, but still the small details I didn't notice earlier, like when he was thinking how he will feed Vanya through the hungry winter, and he said, "I was lucky. I bought 100 of eggs and I bought a pack of apples, so I can give him every day of the winter. I can give him 1 apple and 1 egg, and thus he could survive." And on the February 24, I had very similar thoughts. I ran to the shop, and there were a lot of people already there. A lot of stuff was bought, but I found milk formula, yeah. So to be safe with the child, yes. And the other thing that Boris didn't write, I think, it wasn't in memoir, but when his wife Katya was arrested by Soviet organs, repressive organs, so he could be arrested also because he was a member of the family of the enemy of the state. He could. He doesn't write about it. He writes, so many colleagues were arrested, this and that and that and that, but I believe so these people from this authority, they were coming at night. So at night everybody were afraid to sleep, and there was like lying and listening. And today we spent our nights the same way. Before the going to bed, we are looking where the military drones are going. So the pieces of map are going red because they usually go from south or east. And then there is some message that the strategic bombardment planes, they're started here near Murmansk, and they're going all the way down to the Caspian Sea, and then they're sending their missiles to different parts of Ukraine, and nobody knows which part will be hit today. Yes, and then you're just sitting at 5 o'clock in the morning and looking, yes, because you don't want. So for me, I don't want to rise up my daughter and my wife because they have problems with sleep, and so I need to be in charge. So to put them somewhere in a defended place, and then when it's all over and we're safe, in the morning we read what's happening, how many of them, of these missiles and drones were flying. Even now they draw what are their routes, where did they hit. This is Lviv, where Yuri Saliga, our colleague, lives and works, and this is burning power plant near Kiev today. So it's just happy thing that I have electricity to speak to you. But still, let's come back to Boris Ballinsky. So in 1925, as we heard yesterday, he entered Omelchenko Biological Institute in Kiev. And there he met several guys. One of them was Vladimir Pravdich Neminsky. And Boris writes, so he wasn't a scientist of high repute. No, so this was some guy, he was doing something. And of course, maybe this was the thinking of Schmalkausen himself, and young student just repeated what elder staff is saying. But this Pravdich Neminsky wasn't this simple. Yeah, he was a man who was the first to record EEG like we understand it today, electroencephalography. He put the electrodes not to the naked brain like previous researchers, but to the skull of the animal, of the dog and of the frog, and he found out the same waves we know today, like alpha, beta, gamma, mu, and others. But he called it a little bit differently, electrocerebrography, and different names of the waves, but published it in a pretty good German journal in 1913. Still, he didn't finish his work. He didn't make it on man. And then the German Berger made it 15 years later, but doing the same methodology with string galvanometer, and everything was the same, just he make new names. Of course, Pravdishny-Minsky wanted to object, but he didn't have time. He was arrested and sent to the northern Russia, somewhere near the places the planes now go into Ukraine, and he was persecuted for many years. He survived happily, but still he couldn't defend his priority, and then the Iron Curtain closed in Soviet Union and became the closed state. You couldn't prove anything. Another person from the members of Boris Ballinsky is his aunt Valentina, as she called her everywhere. In the other war years, war and famine, 1920s, 1921, she was trying to do very, very progressive and pioneering work to culture cells, tissues out of the mammal organism. She works with different tissue cells like fibroblasts, like immune cells, like thymus and others, and her work was on impact of pH, how many hydrogen ions can survive the cells outside the organism. And it's one of the, it was published in Journal of Physiology, British Journal of Physiology, and she constructed her own electrode to measure pH in these cells, and she really made possible, was one of them who made possible today's cell culture with these pH indicators. She was also, she was talented researcher in many fields of physiology or biochemistry. She was also arrested like as Ukrainian nationalist because she was patriotic and she was involved in politics during Ukrainian revolution, but then she left it, but still they remembered her and they arrested her for 7 months. She was detained, but then she was released, but couldn't occupy any high positions in Ukraine, and then she emigrated with Germans and died in the United States. But while she wasn't appointed the professor of physiology in medical institute, another researcher the same age as her, Danilo Vorontsov, who is from Belarus but spent most of his life in Ukraine, he came and established a whole research school. What did he do? He measured electric potential. He was classic electrophysiologist like Pravdishny-Minsky. He measured electric potentials of tissues, of muscle tissues, of nervous tissues, and one of the best his results is his and his student Platon Kostyuk. They measured the potentials in to neuron reflex arc in spinal cord. And so their work was noticed by John Eccles, the Nobel Prize winner, and he invited his student Kostyuk to Australia to work with him for a couple of years. And these guys, they established a whole new, for Ukrainian science and for Soviet science, a whole new research school of electrophysiology, of cellular electrophysiology. And they started in the university and then came to Bogomolets Institute of Physiology in National Academy of Sciences. They important breakthrough published in Nature in 1970s. It was very difficult for Soviet scientists to publish in Nature, but they coped with it somehow and published. So they invented completely new method to change ion composition of neuron soma. And before them, most of researchers, they worked with giant squid giant axon, and it's like a tube, and you can do everything with it. But with the whole cell body, which is not round, with all these processes and irregularities, it's quite difficult. And they found the way, so they made small plastic tubes to stuck to the cell membrane, to make a hole and to change ions. And thus they could measure many things and discover a lot of ion channels that are in the membrane of cell. We celebrate 100 years of Platon Kostyuk this year. He was born in August, so maybe some celebration, even despite war, will be held. And his students worked a lot with dorsal root ganglion neurons that are involved in nociceptions, the molecular and cellular correlate to pain. And there's quite a lot of receptors of pain, peripheral receptors, but the main are three. These are TRPV1, acid sensitive and ATP sensitive channels. So TRPV1 is capsaicin, red chili pepper, actin substance receptor and heat receptor. And this is proton receptor and ATP receptor, and these two of three were described by students of Platon Kostyuk. Other stuff his students showed that there are two different types of calcium channels, large conductance and small conductance, transient and long-lasting. Interestingly, that these students of Kostyuk, Krystal and Pidoplechko, Krystal is in Ukraine, he's not very active now, but Pidoplechko is in the United States and he is still working. They were like they stepped the steps of Valentina Radzimovsky. They still go to the pH topic. There's no connection between them. I don't know even if they knew about her. I'm going to interview Volodymyr Pidoplechko, maybe he could tell us. But these idea links, it's fascinating for me. So like in 60 years after these experiments, they discovered these pH sensitive currents. In the very beginning, a lot of people didn't believe them, how the proton concentration can open the gates of ion channel. But still they proved it in accurate experiments, and it is known that in Kiev it was shown. My own research, several words about it. Now looking back, I see it was also connected to the problem of development and the problems that were interested in which Boris Ballinsky was interested. Yes. So my first paper really is an ontogenetic aspect of expression of ion channels. So these are these low threshold calcium channels that were discovered in Bogomolets Institute of Physiology, and there were some works that were studying electrical currents, transmembrane currents, and they showed that these currents are low in the newborn rats, and then they're dynamically increasing during ontogenesis. That some development or post-embryonic development to these channels happened. We looked very primitive, then just common PCR on mRNA level, and saw no difference. So we had different ideas, but then we focused on one of the types of epilepsy. This is absence epilepsy. It's not like in movies like general epilepsy with trembling and everything. It's just the like turning off of the brain or during this several seconds, maybe several dozens of seconds, not very long. And it in humans, it usually happens in children. So when they grow up to adolescence, it is mostly lost. But sometimes, and we don't know why, it turns to be a general epilepsy in the adulthood. So that's why it's medically and socially important. And so we found a rat model, hereditary rat model of this epilepsy, and we start looking what's happening in the young rats, and then in these rats it's vice versa. Young rats are not developing these seizures, but older rats, like 3 months and older, they develop them, and in older rats they show that expression of these calcium channels is growing, and they are one of the suspected players in this epilepsy etiology. And we found different thing, that the currents and the level of mRNA was lower in this animal model, so suggesting there is some regulatory mechanism that plays. And so some of these data we made with my friend and colleague Bijan Sharopov. Unfortunately, he was killed during this war. He was defending the cities of Ukraine. And we found one of the candidates for regulation. We found one of the microRNAs, microRNA1 in rats, that it's probably it can regulate these calcium channels in cortex and thalamus, the important regions for this absence epilepsy development. We predicted them bioinformatically, and then found reverse correlation of expression of this microRNA and one of these calcium subunit. But in the direct, in vitro experiment, we couldn't find it. So maybe there is other players also in this way. And to the end of my talk, I want to emphasize that despite our Institute of Physiology and Department of Kyiv Academic University, where we were working together, and despite we were concentrated historically on electrophysiological methods, ion channel properties, and so on, today it's not the special program, but somehow several groups they turn to problem of neuroregeneration, and of course it's very important problem. During the war, a lot of people have injuries of spinal cord, brain injuries, different ischemias, neuroinflammation, and all this stuff. So it's important to see whether we can revive these patients, and of course we should better understand this mechanism of neural regeneration if it's possible, and in some situation it is possible. And so there's just a couple of examples of research where in organotypic culture the neural stem progenitor cells were taken and put in adult brain slice, which was under ischemia, and the researchers observed how do they how do they get involved in neural networks? How do the electric activity becomes synchronized with the activity of local neurons? How the electric properties changed? And another work is just using adipose-derived stem cells in the same model, but this is a model of neuroinflammation. They can transform to oligodendrocytes to protect and promote axons, neural processes to defend them, and to balance microglia and astrocyte proliferation. And so the results here and here are quite promising. So we Ukrainian neuroscientists, we combine many methods. We're collaborating with the European, American labs, and of course, I think if you are interested, we can find the common points of interest, and we're always open to collaboration because we have financial problems, and to overcome, it's good to overcome it together with other colleagues. We're doing what we can, others doing what they can. Now, these are a couple of my colleagues, and this is Professor Belan, Professor Vaitenko from Kyiv Academic University, and also Bogomolets Institute of Physiology. And this is our histologist, Oleg Tsupikov, who's doing these brilliant masterpieces which win on the science photo competitions. And to the end, I want to thank you all for listening for all these days, to my colleagues for very interesting talks, for several of my colleagues who mentioned me in their talks. Yes, I was really pleased of it. And of course, the main grateful to Neville Pillay for leading all this, and the whole South African team, WITS, and people out of WITS. And of course, Ukrainian team and Alex Gomovsky, who has done majority work on the program, and the Ukrainian embassy in South Africa, who promoted this all this time with all these troubles, but still is with us. And also Vladimir Korsha, I can't avoid thanking him again. I'm very happy to see you, Vladimir, and thank you for everything. Zvimka Kachur, the Ballinsky family, who brought a lot of precious family materials, and some history of science digging was possible due to fellowship from Linda Hall Library in Kansas City, United States, and Ukrainian soldiers who are fighting for me, for sitting here speaking with you. And Bizhan Sharopov, we remember him always. Thank you very much. I don't hear you, sorry.
If anyone wanted to make a comment. there's nothing in the audience and nothing online. Very complicated neurobiology. Some of us are not working in those spaces, especially with gating and channeling and so on. But we did enjoy the history that you wove within your neuroscience background. And Alexei, thank you very much as well for being part of the team that put some of this symposium together. And all the best in the war and the struggles. Thank you very much.
Thanks.
Right, we've come to the end of 2 days. It's been an interesting symposium because it had a different feel altogether from the normal symposia that you would attend. It celebrated Boris Ballinsky as the scientist and had in among it quite a lot of complications with the Ukrainians and the war and so on, and they had to join in online. But I think I would like to first thank the speakers for the session, and then as a general thank you to all speakers that presented yesterday and today. I think we've enjoyed listening to a range of talks today on different sorts of sciences. It certainly tested my range of biology. Most of it I had to dig deep into my undergrad, but I did follow the sorts of general patterns that people are talking about. But what's important is that they all resonate broadly with what Ballinsky would have done in some sense. And you can see some of the sparkle that was started by him that now evolved into all sorts of more complex biology. It would have happened through time and through technology, but you can see how that he had a lot to add to what we know as modern experimental biology. So thank you, everyone, and I'm going to hand over to the Dean for a vote of thanks, but I think we should give ourselves a round of applause first.
So, colleagues, it's a pleasure then for me to close the symposium. I'm sorry that I could not be here for all of the time. Since leaving you yesterday, I will probably sort through 100 emails and attended more than a couple dozen meetings dealing with HR and finance and all of those sorts of things, whilst you are clearly having an awful lot of fun. I am certainly very jealous. It fascinates me that you get out to some interesting locations. I mean, that's the nature of your work. And I certainly appreciated the presentations that I listened into this afternoon. But at the backdrop and the reality is the war in Ukraine, and that's really a tragedy of immense proportions, and it's played itself out in very real ways here with this symposium. Very, very glad that the technologies worked as well as it did. You pay for what you get then, Neville. I queried that with you. I was quite concerned about the costs, but I can see that the quality of the audiovisual technology has just been superb, and so that has meant that we could connect up under these extraordinary circumstances. Colleagues, science brings people together, and that's what's clear about this, and this is really the very hallmark of this Ballinsky Symposium, that despite the fact that we are in the midst of war in the Ukraine, a country that we care for a great deal, a country that we have great admiration for because of its scientific history in particular, but because of the brave and valiant people that stand up for against tyranny. And I, of course, long very much for when thank you. So I long for very much when peace can descend on that piece of land, and when and then I think Ukraine can emerge as a beacon of hope and democracy in that part of the world. But keeping in mind that South Africa and Ukraine were only two countries that voluntarily dismantled nuclear weapons, it does bring home a lot closer the connections that we have with that South Africa has with Ukraine. And so I hope that we can continue to collaborate, and Ballinsky has, over the many years that he was here at WITS many years ago, he still brings us together in this very special way. And I sure hope that this symposium has been the basis for us to continue with our scientific cooperation between both countries. Clearly, there's a lot of science that is happening, not only with WITS, I've noticed with Northwest University, but basically the way in which I see this is that that science is extremely important for the development of both countries. South Africa is still very much in a developing state. And the great traditions of science in Ukraine that go back many centuries must continue. It will be severely problematic if that degenerates through these difficult times, because science is going to be an important means of rebuilding in that country. Just want to share with you that as Vice President of the International Union for Pure and Applied Physics, where I serve as Vice President for the membership portfolio, one of my proudest achievements was incorporating Ukraine into this 100-year-old organization. And 2 years ago, I arranged for the Ukrainian delegation, head of the Ukrainian delegation, to address the General Assembly comprising some 65 countries. And that happened, I think, I attended online, I haven't traveled in a bit, but that took place in Italy. And this year, the General Assembly takes place in China, and I had a word with the ambassador, and I did say that I will try my best through the International Physics Union to ensure that the Ukrainian delegation is well supported in its participation as a fully fledged member at the General Assembly in China. So we will try to fund that. So I'm very keen to see Ukraine succeed and for peace to descend and for us to continue with our scientific cooperation. So colleagues, lots of folk to thank. It really is a great pleasure for me to thank firstly Professor Neville Pillay. Neville, you've been outstanding in your work. I've leaned greatly on you whilst you've had a lot of the documents emblazoned with my name, being the chair of the organizing committee, but in reality you did all of the hard work and you made this a great success, and I really want to express my sincere appreciation to you. So the organizing committee was put together, and it goes to show in this modern technology that we can work well together under tight financial constraints through our hybrid electronic means. So hopefully that is a relatively low energy barrier for us to continue with our engagements. But all of this was organized basically through electronic engagements. But particularly I want to thank Professor Alex Gomovsky and on the Ukrainian side, and Professor Francesca Perini. Is Francesca here? For her assistance, of course, at a time when Neville was not available. So obviously the Vice-Chancellor supported this right from the very outset. First conversations we had about this was about 3 years ago, and of course COVID did not make it very easy, but Professor Vilakazi and myself chatted, and the Vice-Chancellor was very supportive of this right from the get-go. Very grateful for that. And of course, Her Excellency Ambassador Abravitova, I worked very closely with her. I even had tea with her in our office, which was very nice. She very graciously received me at her offices at the embassy in Pretoria, and I do consider her a friend. I've supported many of her activities. I seem to be on the mailing list. Last time it was Ukrainian Independence Day, was it? And we were treated to quite a lovely cultural event in Pretoria. So obviously speakers from both Ukraine and from WITS and beyond. In particular, I was very glad that Professor Hazel Siv was able to join us from Northeastern University in Massachusetts. Most delighted to have seen Professor Barry and Fabian. That was very, very special, and still extremely witty and smart. I appreciate my engagements with him, and also he's reminiscing of the past with Boris Ballinsky. But the many presentations made by all colleagues, and once again, sorry that I could not attend much of it, but certainly that's what's counted toward a successful meeting. I'm sorry that there aren't any proceedings. I get it that in the modern sort of era, one is kind of maybe too busy, or perhaps proceedings of conferences, workshops, perhaps less important now. But still, I sure hope that this will be an important historical record, and perhaps we can look back on this event as a beginning of a new relationship that we have between WITS and in particular the Ukrainian Academy of Sciences. I sure long for us to continue these collaborations. Ms. Zvinka Kachur and Ms. Anastasia Kapranova, two Ukrainians based in South Africa, provided a crucial link between the Ukrainian embassy and the Ukrainian scientists. Thank you very much for that. This ensured that the program itself was a success. And we have gratitude also to James Harrison, Donald McCullum from the Apes Museum. We've seen this magnificent display out in the foyer. That was quite tremendous. I really enjoyed that. So I wish to acknowledge further colleagues from within WITS, Kenrol Magamato, who works from the Vice Chancellor's office. He was certainly an important liaison into the Vice Chancellor's office. And Rochelle Sanker, thank you very much for all of your work. Now that you've sat in through all of the symposia presentations on the subject, you do know you're going to write a test or examination, right? So I'm sure you've paid a lot of attention to. That's what we do. We give tests and exams. We enjoy doing that. And Tembi Dlamini, she and her team. Tembi's always really quite fantastic in helping us organize events. And Sharona Patel, of course, always work in the background and ensuring that things happen. And whenever folk escalate issues to me, I just need to pick up the phone to call Sharona, and I consider that to be fixed. This has been a well-organized and well-run symposium, and I just want to acknowledge that. And I can't thank the AV team sufficiently for the quality of the engagements. And next time, I'm not going to complain about the costs, because you get what you pay for in the end, isn't it, Neville? So, colleagues, thank you once again. It really leaves me then to close the symposium and to wish you all the very best. This is not a once-off event. It would be a shame if it is. I would like to see this being the beginning of a new era of cooperation. Both countries need it. Both countries have a strong yearning for development, and science is a great basis for bringing people together and a basis for people development. Our future is extremely important in both countries, and let's continue to work well with each other. Thank you then. Ciao.
Okay, everyone, we have to conclude. I asked the technical team if they can put everyone online, and we can now see everyone, or most of them, that are online currently, that are sitting around the world. I see a lot of the Ukrainian scientists. I see Denise here. Welcome back, Denise. Vladimir again, and Alex at the top left. And I think we want to share this moment with you. There's a final tribute to Ballinsky and the symposium. And thanks very much for making yourselves available from afar. Okay, and that ends the proceedings.